Yes, sir. So we'll have to ask
You
sir
can
to,
introduce.
yeah. So the live streaming, I'll start.
Manali, you can introduce
Yeah.
Rakesh and
Yes, sir. Yes, sir. Greetings and welcome back to the second session of the Indo-US workshop on recent advances in metabolic engineering for microbial cell factories. We shall start this session with presentations by three scholars from IIT Kharagpur highlighting their PhD project. The first speaker is Mr. R. Rakesh, who is a second year PhD scholar from P.K. Sinha's Center for Bioenergy and Renewables, IIT Kharagpur. He is working under the guidance of Dr. Amit Ghosh. His presentation title is Sustainable Approach for the Treatment of Organic Waste by Integrating Dark Fermentation and Microalgal Culture. Rakesh, please.
Good afternoon to one and all present here. Am I audible to? So like
Uh,
till now, like the workshop was dealt like in designing beta organism, microorganisms using systems biology and synthetic biology. So once you design an organism or microbes, what is the next step? Like you use a specific bioproduct, right? So the bioproduct should be in higher titers. So that can be done by your scale-up process or bioprocess engineering. So in this section, I will be dealing with process engineering approach, as well as I will cover like in the process engineering approach by uh, by treating organic waste. Like so, I will like use organic waste to produce biofuel that like hydrogen and algae. Like so that will be the overall content of this presentation. So let me. So like speaking about this current scenario, like what's happening is that in the current world, like energy, water, and food, all this are like interlinked. Like for what the production of food crops, we need energy, and like from water we can deliver energy. So all this have like having a connection altogether. So like as population is growing, the pressure on all these three aspects is increasing. Like the demand is like very much high on all the process. So ultimately we have pollution and high demand of energy. So pollution arises because like when as the population increases, we have like high amount of vehicles used, and like since vehicles are used, like we have carbon monoxide and greenhouse gases which has been excreted out. And like in the generation of food crops, we have like byproducts like lignocellulosic waste, which has been a major byproduct. And like in water treatment plants also we use high amount of energy. So altogether at last, like we have like rising pollution and high demand of energy. For this, like research have been like diverted to go for alternative fuels, like that is renewable source of energy. So like we have like solar energy, wave energy, geothermal energy, wind energy, and hydro energy. And like I will be focusing on bioenergy. So like since Amit Ghosh sir have already given a introduction on like generations of biofuel, I'll just give you a recap. There are like four generations of biofuels. Let us first generation, second, third, fourth. The first generation deals with your food crops. That is the biofuel fuels has been generated by using your food crops. Like the food crops are like produced like corn or sugar cane, and like starch from potatoes, and like oils from like rapeseed oil and jatropha oil has been used. And like from there we harvest by energy. And in second generation we have used
I'm sorry to interrupt, Rakesh, but it seems they cannot hear you. Someone said in the chat that they want you to raise your voice.
okay. Okay, so what about now? Is it audible now?
Yeah, it's better.
So like in case of second generation, what we use is like we use waste products. So since I've already said like lignocellulosic is a major waste product, and like apart from that we can use manures and like industrial waste. Like extracting energy from the waste product is second generation. And third generation is your like algal fuels, like macroalgae as well as microalgae. Microalgae is like a algae which has been produced like with a small size, and macro seaweeds, all this called as macroalgae. And fourth generation is like which uses advanced engineering principles, like for example all this metabolic engineering approach, all this comes under fourth generation, where we target organisms or metabolic pathway to deliver specific products. Like Amit sir like explained, where like the for the ethanol production, xylose pathways has been like altered so that consumption. All this come under fourth generation of pathways. So like what are the major biofuels which have been present? Like there are like different biofuels which can be are present. That is biomethane, biobutanol, bioethanol, biohydrogen, and biodiesel. So biomethane, everyone would know, like this is a very classic and like day old fuel, like where like even in villages people even now like they use. And like biodiesel like is from where like from oil it has been transesterified and then we get fuels. And bioethanol, where like India is now targeting so that we can increase the blend of ethanol in our in the diesel which has been already used. And biobutanol is like a newly emerging fuel where we no need any no change in engine is being required. And biohydrogen, among all this fuel, like the beauty of the biohydrogen is that it is a only fuel where like after combustion we won't greenhouse the product which is released is not greenhouse gases. So the product of hydrogen is generally water, right? So water is a clean byproduct. So biohydrogen is tagged as clean fuel. So as I've already mentioned, it's a renewable source and non-polluting. And like the other advantages, like it has a major high energy content among all the fuel. Like if one with a small quantity of hydrogen itself, we can extract high amount of energy. And the production process are like thermochemical, electrolysis, and biological. So the starting two process require itself a high energy to produce energy. So which is like which has a drawback. So whereas in biological process we can use mild temperature and like organisms and as well as organic wastes and residues as feedstock. So biological process is preferred. So among the biological process, like there are like a photolysis and like dark fermentation. So dark fermentation is preferred. So like the dark fermentation is nothing but fermentation. So in the coming slides, I will explain what dark fermentation is. So like, and it is an anaerobic process. So in this work, what I have done is like since like organic feedstock has been chosen. So for any if you consider any microbial purification, we need a carbon source, right? So like so here like the organic waste which has been chosen is starchy waste water. So like since it has a high amount of carbohydrate, which is a major carbon source in this waste. And like and like this is the picture of canteen wastewater which like in my work I've like used. And so in starchy wastewater, like the major drawbacks is that it lacks micronutrients and nitrogen source, which is very also important for the proliferation of microbes. So like we want an alternate additional nitrogen source. Adding any ammonium salt or ammonium nitrates is not feasible. So like we have to like go and search for like a nitrogen source which can like fulfill that category. So there are already many nitrogen sources which is existing. Like people use distillery effluent byproducts. Like after like once distillation is done, the rest of the byproduct can be used. Here, like in my work, I've used poultry manure. So why poultry manure? Like since now like people have targeted like use of poultry manure just for like production of biomethane. Okay, so like for like biohydrogen, like it is not produced because like poultry manure has high amount of ammonia in it. So which is a drawback for like hydrogen production itself. So this was targeted like to use like poultry manure to produce biohydrogen. And another major fact is like so India stands for fourth globally in the poultry industries, and GDP around like 4% has been contributed with poultry industry itself. So it's very important to like valorize or like utilize poultry manure for the production of, so that decontamination can be done effectively. So like so in any process, like if you see like any general process, like for example like even in engineering systems biology, in engineering process which you see, we need a feedstock. Okay, before the feedstock we have to select and we have to process it, right? So the preliminary steps are nothing but collection and drying and grinding. So this was the collection and drying which was done poultry manure. For starchy wastewater, like I just collected it and like kept it. And like after once you collect the organic waste, so we have to see what are the what are the other like components which is being present, and the criteria which is are like there in the organic waste. Like for example, TS is nothing but total solids, like whether they have any solids or not. And like TSS is total soluble solids and dissolved solids and volatile solids and volatile suspended solids. These are the characterizations which should be done like if you use waste for production of any bioproduct. And like apart from this organic contents like a carbohydrate, protein, and like has been targeted. This carbohydrate content as such like gives you like the COD content, and the pH like should be measured, since it is a major factor which influences for the microbial proliferation. All are these taken in note. And after this, like if you see any process, like the first step is like we have to see whether the substrate which is being used is like producing the desired product or not, right? Even for example, like once people have engineered microorganism, they use glucose as a medium, like so like how much of the glucose, how much glucose is required, and like what are the quantity of other micronutrients required. So those are called like preliminary stress, and like we have to see whether the organism can utilize glucose itself. So in this case, like since the feedstock is Sachi waste water and poultry manure, I have to see whether the microorganism which I'm using, that is mixed consortia. So I kind of isolated the microorganism from cow dung so that can produce biohydrogen. So that was done, and after that, like the organism which is isolated, I should see whether like the waste which have been targeted can be utilized by this microorganism source. So this is a preliminary test which I used. So like where I've increased the COD content. COD content is nothing but it's a organic content of the feedstock, which can be used for the proliferation of the microbes. Okay, so like COD, the starchy waste water was increased, matlab, sequentially, and like we can see that there is no require, like in the pure starchy waste water itself, like the microorganism can consume like starchy waste water. And in case of poultry manure, like you can see that like, so as the poultry manure concentration increases, like the microorganism proliferation was decreased. So like we have to optimize this ratio of the poultry manure. So then sorry for the disturbance which has been having in the background, like, and so like since like you can see that the proliferation of the biohydrogen is like very less in this case, so we have to add both of the substrate here. So here what happens like the poultry manure and the starchy waste water was mixed. So like here the percentage of the starchy waste water and was kept constant, since starchy waste water as such can be utilized uh, by the microorganisms, whereas the poultry manure was optimized increasingly, like with using 1%, 2%, 3%, 4%, 5%. So this is called a single parameter optimization for the production of biohydrogen. So the experimental condition has been mentioned, like I've used like 30 ml like serum bottle, like this is basically just like a batch culture, like where people use shake flask for the production of a bioproduct. Here, like since I've already mentioned you, like biohydrogen is a dark fermentation process, so we need an anaerobic condition I mentioned, right? So this so we need a sealed environment, so this can be got by using serum bottles here. So like serum bottles can produce anaerobic environment easily, and before that we sparge nitrogen and all to produce anaerobic. These are the like very next matlab, aspects which you have to know. As of now, you could see this anaerobic condition can be maintained here, and then mixed consortia is added in the condition which is being used. So like apart from single parameter optimization, we can also use multi-parameter optimization, where like the like like different concentration can be maintained, as well as the process parameters like pH, reaction temperature, inoculum age, different can be done. Like this can be done by computationally, and like later we can fix them from value. So these are the two conditions which is important, like if you like target any bioproduct to produce. And next what happens is like, since like I've like designed it in a preliminary stage in small scale, so we have to scale up the process, right? Like if you like produce it in a test tube, then you go for shake flask, and like after shake flask you have to go for reactor level. So here, like since I've like done it in the preliminary studies in the 30 ml reactor, so I then targeted to like 500 ml reactor. So this is just a basic laboratory setup where like you can see two reactors here. These are like double jacket reactors, we call them double jacket reactor, and there's a cork here like fixed. Since it's an anaerobic environment, so we have to close it so that no oxygen can be incorporating. And like you can see the water inlet and outlet to maintain the overall temperature. The temperature is 37 degrees Celsius. So here this is a setup of hydrogen where like gas can be produced here and can go through a separation column. And since like any process here, it's like you have a microbial culture in the system, and they can feed on organic waste, right? So at last what happens is like the product which is delivered is not pure hydrogen. We have like mixtures of other compounds like CO2 and all. So we have to remove the CO2. So this is called a CO2 scrubber, where like NaOH has added and CO2 is removed, and the remaining part is like the hydrogen, which can be measured by using like water displacement method. So where like you can see as the time increases, like your hydrogen is produced. And here like you can see that within the starting period of the time, like there's an increase in soluble carbohydrate. This is nothing but like the solid substance which is present can be are like hydrolyzed effectively by the organism, and there's an increase in the like soluble substrate. So and then like after like hydrogen has been produced, so like the remaining spent wash, that is the remnant, the gas has been produced, and like you have the remaining content in the reactor, right? So they are called the spent wash here. So this has to be like characterized, so like uh, so that when you deliver it outside, like in the running stream of water, like you have to see whether like it is according to the norms of like and like the reduced COD, or like content has been reduced. So in laboratory scale, like these concerns are like not doesn't matter much, whereas if you go for large scale like production of any biofuel or any bioproduct, we have to keep in mind that like the content which we release to environment should be like remediated properly, effectively, so that like it is like not affecting the environment. So contents like nitrate, phosphate, and ammonium was targeted. And like see, biohydrogen product process is a process where like we have a volatile fatty acid production. So we have volatile fatty acid like butyrate and acetate, which was maximum produced, and we can't release this content as such to the environment. So like remediation is important here. So till now, people have explored utilizing butyrate and acetate to produce methane. So if you see in the methane production, like the preliminary step is like consumption of volatile fatty acid to produce methane. So like, but methane is, since I've already told, is a day old technique. Simple, like we can use microbial algal feedstock, or like to utilize this fatty acid, volatile fatty acid. And as already Piyush mentioned is in the slide that engineering of microalgae can be done to produce various bioproduct. Here it's clearly shown that from algae we can produce bioproducts like biopolymers, pigments, and different pigments which are like very therapeutically valued, and feedstock, biofertilizers, and biopharmaceuticals. It can be produce biopharmaceutical products also. Like like after this, so like, so in microalgal, like the growth of the microalgae happens with three different stages. Like there are like autotrophic growth and mixotrophic growth and heterotrophic growth. So in my experiment, like what I chosen is mixotrophic growth, where like I will sparge CO2 as well as I will use inorganic carbon sources, that is your acetate and remaining carbon sources. So what is autotrophic growth is nothing but like just general photosynthesis, where you use sunlight and your carbon dioxide for the proliferation of microbes, growth of cultivation of microbes. In heterotrophic, you don't give your sunlight, whereas it is a dark process, and we use only like organic content which is present, and like accordingly like we produce our microalgal cultivation. In mixotrophic, as the name suggests itself, use both the condition, like your soluble organic content as well as your like CO2 content. So like, for producing microalgal cultivation. So like if you see in general, there are like different microalgal species which has been present, like neochlorosis, chlorophyll, chlamydomonas, sandices, and genella. All these species can have like different carbohydrate content and lipid content and protein content, and like doubling time of like different. And like in my experiment, I chose like chlamydomonas randati, like since like the major advantage is that it has a very less doubling time, and it can have like additional pigments also. So like, see, doubling time is a factor which should be kept in mind, because like it requires only very less time to like produce like to from one concentration to another concentration, double the concentration, simply the name suggests. So this, if the doubling time is less, so within less period of time we can get major biomass, right? So this is major advantage here. And apart from this, we had like both lipid and carbohydrate. And one more major advantage is like the whole genome sequence is already available here. So my colleague was working with in manipulating like genome of chlamydomonas, so that she can flocculate chlamydomonas itself. So flocculation is nothing but you just clump up your microalgal cultivation, cultivated microalgae. So that is a major advantage, where like separation, centrifugation of algae as such can be are done, which is a very hectic and cumbersome process. So if you use self flocculation itself, like, so that can be an advantage, right? So like my colleague was working with that. So suitable concentration, like, so what happens is like once you select your organism, like I have selected chlamydomonas, right? So we have to like again fix a concentration here, like that is again comes under single parameter optimization or batch optimization, where like I kind of range with different concentration of your spent wash, like 25, 50, 75, 100. Like with the pure like spent wash without mixing with any tap water, there was no growth in like after 50, 75.
Rakesh, sorry, a little bit louder.
Yeah. So after like 50, 75, and 100 percentage, like there wasn't any growth of microalgal cultivation. Only I could see growth in 25% of the concentration, volume per volume, where like 25 ml of spent wash was mixed with 75 ml of water. So that is like 25 percentage. So like can be given. And I could see that like the biomass, carbohydrate, and the lipid content of the cultivated algae was 1.45 gram per liter, which is really a good value. And like carbohydrate and lipid content with equal amount of values. So like then this was like preliminary steps which I did like with a batch process with a volume of 150 ml. So I have to scale up again, like whether we have to see whether the same value is coming up or not. So this is like a next step where like I scaled up using a airlift reactor, like a volume of 1.2 liter, like using the same algal species with a vinoculum volume of 10%. Whereas if you see in other in industrial applications, people use raceway pond as a major process. But the like here you can't you we cannot use raceway pond as in process, because it here we give an organic substrate, right? Like we give acetane and butyrate. So it should be a closed chamber, because like these organic substrates are very much susceptible to get contaminated by bacterial species, or even yeast can utilize all this acetate and butyrate to produce. So we have to keep the systems sterile. So we use closed system like photobioreactors or LED based bioreactors for this remediation of spent wash. If we opt for if we opt for biohydrogen production in the first stage. And like here like now once like you see once you cultivate the algae, we have to see whether the performance of the algal production, like whether the bioremediation is happening or not. That is nothing but whether the media which you give is been remediated or like or consumed by your any microbial species. Even back then you would have seen that in for the production of biohydrogen, we saw like the time scale and everything aspects, right? So here if you see, as since like Chlamydomonas had 6 days of growth growth condition, so it was taken till 6 days. And like the parameters like biomass and time was analyzed. And apart from this, like consumption of COD was also seen. So you can see that the COD is gradually reducing. That is, COD is nothing but, as I've already mentioned, it's the organic content which is present in your spent wash, right? So as you like microalgae growth grows, you can see the COD is reduced and biomass is increasing. And here you can see that pH is increasing. So the algal has a condition of like what happens is, we doing algal cultivation is, since it utilizes CO2 or CO2. So like the CO2 gets mixed with water. So automatically in the first period of time the pH will be less. And like as the pH is like consumed like more and more, like the pH is like increased. So this is what it shows, like pH is increased here. And like here like the other content like nitrate and phosphate and ammonia, since I mentioned already, these are the major content which should be mentioned to see whether like the water is quality enough to be left out in the like stream or like in the river stream. So these are the mentioned. So you can see that as the time interval goes, like the content were reduced effectively. And like, so what, I cultivated the algae, right? So what is the next step? Like unless and until I get a product, what is the use of it? So like here like since like the target product is biodiesel. So how biodiesel is produced is, right, like from fatty acids, that is triglycerides. Okay, and like you need a catalyst here, and like you need a solvent, methanol. And like after this reaction, whole reaction is called as trans-esterification reaction. So after so with the trans-esterification reaction, we get a product of biodiesel. So this was later done like in my experiment. And this setup is like where you extract your like lipid content using your methanol solvent is been will be given here in the setup. And like biomass will be kept here. And like your like reflex will be happening, 3 to 4 reflex. This is called a Bayer method. And like you use methanolic HCl as catalyst, as I mentioned, catalyst here. And then you get a product here, biodiesel, which will be measured by using apparatus like GC and all, like, to see whether like what are the content or the content present in the fatty acid, whether the fatty acid is like, sorry, the biodiesel is quality enough to be used, or like what are the different esters which is present can be seen by GC. And like after this, the overall process should be seen from the first stage. This is like the media, like that is nothing but the feedstock which I selected. Here in my case, it will be like starchy wastewater and like poultry manure. If you use another feedstock, you have to keep this as a constant. So since I've used two steps here, like dark fermentation and final effluent, like this is nothing but the effluent which is released after microalgal cultivation. So two steps here. So like all this the steps where like we can see that as the stages increases, we can reduce the COD content, as well as we can reduce the phosphate, ammonium nitrate, everything like here. So it's shown that like at the final stage 98% of the COD has been released, but can be was removed from the initial substrate. In the first stage, it was like 40 percentage, and second stage it was 69 percentage. So like after this, we have to see for any process which deals with bioenergy or energy process, we have to see whether this process is feasible to deliver energy or not. Like so here you can see in the first stage, like 8% of the energy was recovered like from the feedstock itself, and like second stage 9% of the energy recovered. And like in the when you club two processes together, we can get like 18 percentage of the energy which is recovered. Like as such for biohydrogen production, like 18 percentage is a good amount, like using organic waste. And the calorie and this energy content can be calculated by using the calorific value of hydrogen and like calorific value of the biodiesel and all. So if you like target any biofuels, like you have to do this step, which is very important, like to analyze the bioenergy. And then the overall process design, like where like from first stage, like in the by dark fermentation to like the water which has been released out, and the byproduct which is like here like see in the first stage what happens is the organic waste, the COD is was like 100%, which has been given, and which produces an hydrogen. And after this, the effluent which is released was like separated, right? So since it's a solid the substrate, a solid substrate, we have to remove the mixed solid content. So that was removed, and this was seen whether this can be used as a byproduct as a manure or not. So like the quantity of TKN was seen in my experiment. And then after this, like the soluble content can be utilized for microbial cultivation here, which I've used here, since I've already shown can be used for methane production or something. Since since here in this step we required an optimization where like 1/3 was only used, the remaining content was mixed with water here. So like this shows that water has been here mixed here, and after this microalgal cultivation happens, and you have your product which is algae and continuously mentioned, and like the remaining product can be treated water has a COD of 1.8 gram. So I've already mentioned like 98% of the COD was removed in this whole when you incorporate two-stage processes here. If you like incorporate only 1%, like the COD is like 60% was only included. So like integrating the two process, it's very much efficient here. So like here, from this I've come to the summary where like I can say that biohydrogen production was improved in threefold when I used poultry manure as my feed co-substrate with my starchy based water. And I can tell that the algal biomass with high metabolite concentration, that is carbohydrate and lipid, was produced using the spent wash of the after like dark fermentation. And overall process, which is like 98% was seen with a recovery of 18%. So like finally I can say that this integrating two processes, we can produce two different fuels, and like as well as remediate waste, which can happen simultaneously. So with this work, I finished my talk. Thank you. If you have any questions, I can.
Thank you, Rokesh, for your nice talk. A lot of people have actually said that your work was very good, and we don't have questions.
Okay.
So I think we'll move on to the next speaker. Thank you very much. So the second speaker is Mr. Shayak Mitra, who is a second-year PhD scholar from Department of Biotechnology under the joint supervision of Dr. Ridhiman Dhar and Professor Ramkrishna Sen. He will talk on metabolic engineering approaches for improved bacterial lipopeptide production. Shayak, please.
Hello, am I audible?
Yes, you are.
Okay, so I'll share my screen. Is the screen visible?
Yes.
All right, so Thank you for that introduction. I'm Shayok Mitra, and today I'll be talking about basically a perspective on how metabolic engineering approaches can be used to improve bacterial lipopeptide production. So, what are these lipopeptides? They are a group of bacterial biosurfactants. These biosurfactants are amphiphilic molecules with a hydrophobic and hydrophilic moiety. And because of their amphiphilic nature, they can reduce the surface and interfacial tension, enhance emulsification, and increase solubility of immiscible substrates. So, these are produced by bacteria as secondary metabolites, basically, in most cases, to feed on water immiscible substrates by means of emulsification. They also play roles in when it comes to biofilm formation, root colonization, basically adhesion of cells to any interface or any solid surface. Moreover, these biosurfactants, particularly lipopeptides, have well-known antimicrobial properties as well. So lipopeptides have a structure like this, where the hydrophilic moiety consists of a peptide ring attached to a fatty acyl carbon chain, which is the hydrophobic moiety. The lipopeptides produced by Bacillus subtilis can be, in general, grouped into three families, such as iturin, surfactant, and fengicin. So the unique thing about lipopeptides is that because of their cyclic and horse saddle conformation, they're very effective in reducing the interfacial tension. So these are some of the examples of the various lipopeptides belonging to each of the families produced by Bacillus strains. Why lipopeptides? Why are we so interested in lipopeptides? They have been seen to have many advantages over chemical surfactants, which are derived from the fast depleting reserves, fast depleting petroleum reserves. So they are biodegradable. They have low toxicity. They can be produced from cheap raw materials and renewable resources. More importantly, they have specific action, and they have high pH, temperature, and ionic strength tolerance when compared to chemical surfactants. But most importantly, they can rival chemical surfactants in terms of efficiency and effectiveness. For example, the most well-known lipopeptide known is surfactant, which has the potential to reduce the surface tension of water from 72 millinewton per meter to 27 millinewton per meter, which has not yet been matched by any chemically derived surfactant. And because of these advantages of lipopeptides, they are used in a variety of industries, such as agriculture. They are used in laundry detergent, biopesticides. In medical fields, they are used. Their uses are quite well documented, even in food processing industry. And in recent years, their potential use in microbial enhanced oil recovery is being explored. But despite these their advantages and widespread uses, there comes an obstacle when we want to scale up the production process of lipopeptides, particularly because of the poor yields from the raw substrate materials, and also because of extensive losses when it comes to downstream processing, when it comes to product recovery and purification. And all these factors lead to large capital investments. So how can we improve the yield of lipopeptides from bacteria in a large scale production process? The first step is usually the isolation of an efficient producer strain. After that, we go for optimization of physicochemical parameters. This can include medium composition, pH, temperature, addition rate, agitation rate, any other process parameters. But there comes a limit in optimization, where the bacteria will produce the lipopeptides just as much as they need it. They will not go beyond that point, and that is stopped by various feedback repression and feedback inhibition mechanisms. So to circumvent those feedback inhibition and repression mechanisms, we go for strain engineering and optimization, where the genome of this bacterial strain is manipulated. And there comes the various approaches actually summarized by the theme of this workshop, the design of microbial cell factories. These unique unicellular organisms can be trained to take in the raw substrate materials that we provide, so that it can be processed to the target, to the product that we desire. And just like in any chemical process plant, the pipelines of flow of the raw materials and products needs to be precisely designed. When it comes to design of microbial cell factories, we need to precisely design the flow of the carbon and nitrogen fluxes obtained from the catabolism of the substrate material that we provide, so that these fluxes can be streamlined towards our desired product. But here comes the challenge that the cell needs a major proportion of these fluxes to grow and even more to survive and replicate. And herein lies the basic challenge of metabolic engineering, to balance the flux distribution between growth and production. Now, before we dive into how this balance is achieved or how the microbial cell factories can be designed, it is worthwhile to look into how lipopeptides are synthesized by the cells. So lipopeptides fall under a group of peptides known as non-ribosomal peptides. The peptide ring of these lipopeptides is not synthesized by the cell's ribosomal machinery. It is independent of an mRNA template. Instead, they are synthesized by non-ribosomal peptide synthetases, which are multimodular enzyme complexes. This form of synthesis allows for the incorporation of both D and L forms of the 20 standard amino acids, along with various non-proteinogenic amino acids such as ornithine, amino acids, and various hydroxy acids. The non-ribosomal peptide synthetases, as I said, they are multimodular enzyme complexes, and each module is responsible for the incorporation of a single amino acid in the peptide chain or the peptide ring. Each module is further subdivided into various domains, such as the activation domain or A domain, which recognizes and activates the amino acid to be incorporated in the peptide chain. Then the condensation domain or the C domain and the thiolation or T domain work together to incorporate that activated amino acid into the peptide chain. Some modules contain an epimerization domain, which convert the amino acid from the L form to the D form prior to incorporation. And then comes the last module has a domain called the thioesterase domain or the TE domain, which is responsible for cleaving the nascent peptide from the enzyme complex. So first, the adenylation domain or the activation domain A domain adenylates the recognized amino acid. They recognize the suitable amino acid, and this domain has a unique amino acid sequence which acts as kind of a codon for recognizing the corresponding amino acid. They are activated by addition of an adenyl group. Then the thiolation domain contains a 4' phosphopentathinyl cofactor, which is denoted as SH out here. So the activated amino acid or the adenylated amino acid then binds to this particular cofactor, the SH cofactor of the thiolation domain. Then the condensation domain creates a peptide bond between the amino acids attached to two adjacent thiolation domains. And finally, the thioesterase domain cleaves this peptide from the enzyme complex. So this picture actually summarizes how surfactant is synthesized, the stepwise synthesis of surfactant by its corresponding non-ribosomal peptide synthetase or surfactant synthetase. You can see here that in the first module itself, there is a fatty acyl moiety. So the first module itself is responsible for the lipoinitiation reaction or the attachment of the fatty acid chain to the amino acid. One interesting thing about the genes corresponding to these non-ribosomal peptide synthetases is that they are placed as gene clusters or operons under the control of a single promoter in bacteria. Now, how is the expression of these genes controlled? The expression of the genes for non-ribosomal peptide synthetases is under the strict control of quorum sensing system. This quorum sensing system is a cell-to-cell communication system which allows bacteria to coordinate with each other properly. And so, due to this, they can exhibit various social behaviors such as biofilm formation, swarming and sliding motility, and exoprotease production. The basic flow of a quorum sensing system is that an autoinducer is secreted by the bacteria. Now, at low cell density, the autoinducer is present at quite dilute concentrations in the extracellular medium. As cell grows, as the number of cell increases, more and more autoinducer is secreted into the extracellular medium. And at high cell density, once a threshold concentration is crossed, the autoinducer will bind to its cognate receptor protein, which will then activate the downstream signaling cascades for regulation of the expression of the corresponding gene. So here I've taken the example of surfactant synthetase, which is encoded by the SRFA operon in Bacillus subtilis. The autoinducer in this case is COMX, which binds to COMP when it crosses the threshold concentration, and that leads to phosphorylation of the regulator protein called COM-A. COM-A binds to the surfactant operon, the SRFA operon, and this leads to the synthesis of surfactant synthetase. And finally, the surfactant is produced from amino acids and fatty acids. The binding of COM-A, the phosphorylated COM-A to the SRFA operon is actually inhibited by various regulators called COD-Y, SPX, RAPC. Now, how are these inhibited? These are inhibited by something called as the CSF, or competence and sporulation factor, which is synthesized by the PHRC gene. Once surfactant is released into the extracellular medium, it kind of induces a leakage of potassium ions, which then goes on to activate the KIN-A or SPO0F phosphorylase system. The end result of this entire phosphorylase system is the secretion of biofilm matrix components into the medium, which allows for the cell population to form a biofilm, and also the induction of the SPO0H gene, which codes for the sigma H subunit of RNA polymerase, which is specific for the PHRC gene, which again codes for CSF, and CSF, once activated, enhances the expression of the SRFA operon and increases the biosynthesis of surfactant. Work by Z et al. They basically analyzed the genome and transcriptome of Bacillus amyloliquefaciens, which has been proven to be a hyperproducer of surfactant. Their work showed that these are the regulators which act positively to induce the SRFA operon. These include the COM-A, as we discussed, the DEGU, which is a global pleiotropic regulator, the sigma A subunit of RNA polymerase, the global regulator ABRB, and another regulator called RGHR. And on the other hand, these are the regulators, some of which we have discussed in the previous slide. These are responsible for inhibiting the binding of COM-A, phosphorylated COM-A, to the SRFA operon. Now we go into how the strains can be improved, or the strains can be optimized for enhancing the lipopeptide production. One of the most common methods is engineering the transcription. How is that achieved? By the promoter of SRFA operon is an auto-inducible promoter, because it is under the control of the quorum sensing system, which is in which an auto-inducer is released, as we have discussed in the previous slides. So here, the auto-inducible promoter can be replaced with either an inducible promoter, constitutive promoter, or synthetic promoter. The problem with an inducible promoter is that we have to factor in the cost of the inducer, which sometimes becomes a costly affair. Constitutive promoters, on the other hand, have another problem. While they are very good in this to achieve the target out here, but the majority of the cell's proteome becomes, the majority of the cell's proteome is filled with the protein of we are trying to overexpress, which in turn causes a metabolic burden. Next, we can also diversify the promoter sequences by either random mutagenesis or site-directed mutagenesis of the consensus promoter sequence. We can randomize the non-conserved region sequences, design hybrid promoters, or even modify the transcription factor binding sites or alter the ribosome binding sites of the promoter. Next approach is engineering the transcriptional regulators. These transcriptional regulators are responsible for mediating the effect of environmental conditions and simultaneously correspondingly regulating the gene expression. So we can go for overexpression of positive regulators. These positive regulators may include COM-A, PHRC, or the various other positive regulators that have been discussed previously. Or we can even go for deletion of the negative regulators. Most of these transcriptional regulators have an autoregulatory motive, where they either where they activate their own expression so that more and more of these regulators are formed and more regulators can go and bind to the target gene. So this autoregulatory motif can be targeted to modify the transcriptional regulation process. We can also go for manipulation of the pleiotropic or global regulators. Pleiotropic regulators are responsible for the expression of multiple genes at once, while global regulators are responsible for mediating an entire metabolic pathway, like the carbon catabolite repression is mediated by global regulators. Now, one advantage of engineering transcriptional regulators is that it can be used to activate silent gene clusters. Silent gene clusters are basically gene clusters which are not expressed under normal conditions by the cell. So they remain silent, and their protein products are most often not realized until the genome is analyzed carefully. So one study I'd like to draw your attention to is a transcription factor decoy strategy by Wang et al., in which the sequence to which the repressor binds, the repressor protein binds, has been chemically synthesized and cloned into multi-copy plasmids. These multi-copy plasmids were then introduced into the cell, and these plasmids replicated inside the cell. And what happened as a result is that the repressor, instead of binding to the target gene's promoter, they bound in a competition effect, they found binding to the plasmid sequences more favorable. And as a result, with no repressor to bind to the silent gene cluster's promoter, these gene clusters became activated. Now, as I had discussed before, the major challenge of metabolic engineering is to balance the fluxes between growth and production. One way to achieve this is static balancing, where we go for a middle ground, where both are compromised and ultimately a constant value in between is maintained. Another method is dynamic balancing, where at low cell densities, the fluxes are diverted towards primary metabolism required for cell growth and cell division. And at high cell densities, there a switch occurs where the fluxes are diverted towards secondary metabolic pathways. And this can be done in either pathway-dependent manner or pathway-independent manner. In a pathway-dependent manner, the metabolite biosensor transcription factors can be developed. The transcription factors can be engineered to sense the levels of metabolites and direct the fluxes accordingly. In the pathway-independent manner, the quorum sensing circuits can be engineered. So the way to engineer the quorum sensing systems can be many. The most common ones include modifying the regulator, where the transcription of the the regulator protein can be manipulated, or its sensitivity to the signal can be manipulated as well. The next is the modification of the signal uptake. So the signal needs to enter the cell and bind to its receptor protein, to its cognate protein. And so we can either increase or decrease the uptake of these signals by the bacteria by overexpressing or deletion of the transmembrane receptor proteins or the transmembrane channel. Another way is to modify the quorum sensing promoter. So the promoter of the target gene can be modified so that it can either bind more tightly to the response protein, the regulatory protein, or it cannot recognize the regulatory protein at all. And the last method is adding additional circuitry. Next, we go to metabolic flux optimization, where we need to basically rewire or redistribute the fluxes towards the various pathways. The basic sense of metabolic flux optimization involves enhancing the supply of precursors, removing feedback inhibition, blocking competitive pathways so that the fluxes are not diverted towards other products, and increasing the rate limiting steps to enhance the yield further. The goals of these metabolic engineering strategies include enhancement of product yield and productivity. It can also be used to extend range. So in order to get more yield on cheaper raw substrate materials, we can further train the bacteria by means of metabolic engineering to take in cheaper substrates. We can also extend the spectrum of our products and find out novel products or even design novel products as such, and improve cellular properties. Here, cellular properties refers to the tolerance of the cell to these lipopeptides produced, particularly the efflux of these lipopeptides into the extracellular medium. Now, one increasingly popular approach for metabolic flux optimization is pathway modularization, where individual biosynthetic pathways are grouped into distinct modules. Now, when it comes to engineering the pathway, engineering the entire biosynthetic pathway, we can manipulate each of these modules separately in order to achieve our target goal, in order to achieve our holistic goal of enhanced target product yield. So, in the work by Z et al. That had been shown in an earlier slide, they divided the biosynthetic pathway of surfactant into 6 modules. The first being the influx of carbon source. Second is the NADPH generation via the pentose phosphate pathway or hexose monophosphate shunt. This NADPH is an important cofactor when it comes to biosynthetic pathways. Then comes the precursor pool, which includes the biosynthesis of the amino acids for the peptide ring, such as glutamate, valine, aspartate, and leucine, biosynthesis of the fatty acid chain. Then comes the actual work done by the surfactant synthetase, which is biosynthesis of surfactant. And finally, the surfactant resistance genes, where the surfactant synthesized is effluxed or released into the extracellular medium. I would like to draw your attention to one striking example of the use of this pathway modularization approach done by Wu et al. This is a study published in the journal Metabolic Engineering and has garnered appreciable, significant appreciation and interest. Here, the strain Bacillus subtilis 168 initially produced 0.4 gram per liter of surfactant. Next, they deleted the genes responsible for biofilm formation and the genes for other non-ribosomal peptide synthetases such as fengycin and bacillactin and bacitracin. And this was done in order to reduce the competition for the energy source, for the energy substrates or the cofactors for other non-ribosomal peptides or for biofilm formation. They need to be solely concentrated towards the synthesis of surfactant. With that, they obtained a 3.3-fold increase in the yield of surfactant. Then they overexpressed the efflux protein or the transporter proteins such as SWRC, LIA, and ACRB, which improved the cellular tolerance towards the synthesized surfactant. Then they proceeded to increase the supply of precursor branched chain fatty acids. And finally, they enhanced the SRFA operon transcription by diverting the acetyl coenzyme A away from the cell growth. So as had been discussed in the talk by Professor Tang in the morning, there is a push and pull strategy when it comes to metabolic flux optimization, where we increase the metabolic flux towards acetyl-CoA, which is the push, so that because acetyl-CoA acts as a branch point where the catabolic and anabolic pathways converge. And then we pull the acetyl-CoA fluxes towards the biosynthetic pathways of our interest instead of it being diverted towards other pathways. This is the pull of this push and pull strategy. And with this, they finally obtained 12.8 gram per liter of surfactant, which is a significant increase from 0.4 gram per liter. Another study which utilized this modular approach is for the production of iturin, where Bacillus amyloliquefaciens, LL3, the iturin operon was placed under the control of a constitutive promoter, C2 up, and with that, they obtained 37.35 mg per liter of iturin A. Then they proceeded to optimize the fermentation medium using response surface methodology that further increased the yield of iturin A. And finally, they overexpressed the pleiotropic regulator DEG-Q. And with that, they obtained a yield of 1 mg per liter of iturin A. Now, the approaches discussed previously are the more targeted approaches of metabolic flux optimization. Another approach that is used is the evolutionary engineering, where we mimic the natural process of evolution, of generation of variation, and subsequent selection based on suitable environmental conditions. So here we basically we expose the strain, the bacterial strain, to various mutagens, or some other way we generate diversity in its genome sequence, and then we screen for which mutations, which variants are important to us, are can be used by us for our target, are suitable for our target, and for that we use a selection pressure. And finally, we amplify that particular strain, we amplify that particular gene, or we use that particular strain further. And this cycle goes on and on. This can be done in the level of protein by means of directed evolution, or it can be done at the level of the bacterial strain itself. One increasingly popular method of generating the variants is known as genome shuffling, which is basically a variant of the well-known protoplasm fusion method. Here, we have the initial strain is subjected to a mutagen, or which most commonly nitrosoguanidin, and that leads to a parental library of variants. Then the protoplasts of these variants are generated, and these are fused so that the genomes of the variants can recombine to form an even better strain, or an even improved strain. And finally, the selection is done using a suitable selection pressure to obtain the desired phenotype. When it comes to the selection pressure, one popular method of selecting for variants is by creating analog resistant mutants. This is commonly used in case of secondary metabolites. So here, as can be seen in this example, say aspartate is converted to aspartyl phosphate, which is further converted to aspartate semialdehyde, and finally to threonine. Aspartate semialdehyde is further converted to lysine. Now, both threonine and lysine exert feedback inhibition upon the enzyme that catalyzes the conversion of aspartate to aspartyl phosphate. By means of random mutagenesis, we can create a mutant and further screen for the mutant using several techniques, where this particular enzyme does not recognize the analog of lysine or the analog of threonine. Analogs are compounds that are structurally similar to the corresponding parent compound. In this case, the lysine analog would be structurally similar to lysine. So an enzyme which does not recognize which does not bind to lysine analog will most definitely be resistant to the feedback inhibition by lysine. Same goes for the feedback inhibition exerted by threonine as well. And in that way, if this feedback inhibition mechanisms are removed, that way the end product can be overproduced. The approaches we have discussed above are mostly do not give a holistic perspective of the entire metabolic network. To gain a holistic perspective of the metabolic network, we go for systems level analysis, which includes genome scale metabolic reconstruction, which is a form of genome annotation, basically, where the entire genome sequence of the strain is obtained and the metabolic genes are correlated with the reaction, the biochemical reaction that they catalyze. And in this way, metabolic genes can be correlated with metabolic pathways. This is important. Gives significant help in facilitating metabolic engineering of the organism. We can easily design a metabolic engineering strategy using these genome scale metabolic networks, and we can even predict the impact of genetic and environmental perturbations on growth and chemical production. Once the genome scale metabolic network is reconstructed, the next step is the flux balance analysis. So in flux balance analysis, we basically mathematically represent the metabolic reactions in terms of stoichiometric matrices and rate matrices. And then we go for developing a system of equations which act as constraints to these matrix equations. And then, since the number of metabolites always exceeds the number of reactions, we have an underdetermined system which can be solved by means of these constraints, as has been shown here, and also by defining an objective function. More often than not, the objective function is the optimization of optimization of biomass or increasing the biomass. But in the case of lipopeptide production, the optimization, the objective function would be enhancement of the lipopeptide production. That would be our objective function here. And using the constraints and the objective function, we can go for a linear programming problem to basically define a solution space or define an optimal point optimal point which represents the optimal flux distribution. Using this approach, a flux map can be generated, as has been done for calcium-dependent antibiotic produced by Streptomyces coelicolor. This flux map basically gives the rate of each biochemical reaction, and in that way we can see how each metabolite, to what extent is each metabolite distributed in competing pathways, and we can correspondingly inhibit or overexpress the pathways accordingly to streamline our flux pipelines. Apart from this, metagenomics has an important role to play when it comes to lipopeptide production. So the lipopeptide repertoire that we know now, that we are aware of as of now, is not very comparable to the actual repertoire that is actually present in nature. What we know now is from the organisms that we can culture and assaying the products that they produce. Metagenomics comes into play in the sense that we can isolate samples from different environments and extract the DNA from these samples. And after sequencing these DNAs, after analyzing these sequences, we get to know what other secondary metabolites, what are the lipopeptides in particular, they have the environment has the repertoire for. And in that way, we can increase our library. These genes can be cloned, and these genes can be amplified and cloned into the culturable organisms so that we can produce these novel lipopeptides. So to conclude, I would like to say that metabolic engineering is not a standalone field. It requires the help of organelle engineering, synthetic biology, systems biology, structural biology, and in recent years, machine learning and other computational tools are also coming into play to automate these metabolic engineering programs, or to facilitate these metabolic engineering programs for ease of production or to enhance the yield. So to conclude, all these fields need to come together, to collaborate together, to enhance the field of metabolic engineering, or to realize our goal of designing microbial cell factories. So I'd like to acknowledge and express my eternal gratitude towards my supervisors, Professor Ramkrishna Sen and Dr. Ridhiman Dhar, for their guidance and motivation for this presentation. I would also like to take the opportunity to thank IIT Kharagpur, Department of Biotechnology, and School of Energy Science and Engineering, and also the conveners and organizers of the Indo-US Spark Workshop for giving me the opportunity to present this review today. These are some of the references that I have used for this study. So thank you.
Thank you, Shayok, for your excellent talk. Some people have said that your presentation was nice. And we have a question from Rutuja Jagtap. She asks, how about temperature-inducible promoters? Like that's...
Right. So thank you for the question. Temperature-inducible promoters are also being used when it comes to lipopeptide production, but they haven't gained that much attention, or they haven't gained that much popularity. The major problem of temperature-inducible promoters is that a quick change in the temperature is often difficult to achieve in large-scale fermentation processes. So that kind of limits their use when it comes to industrial scale production. So.
Okay, thank you. So thank you, Shayok. We'll move on to the third presenter. So the third student speaking is Ms. Shreya Raut, who is a third-year PhD scholar from Department of Biotechnology under the supervision of Dr. Ridhiman Dhar. Her topic of presentation is how laboratory evolution can benefit metabolic engineering. Shreya, please.
Hi. Yeah, so good afternoon to all. I'm Shreya Raut, and I will be presenting on the potential of laboratory evolution with respect to metabolic engineering. Shayok touched upon it a little bit, and I will be elaborating on that. Can you see me?
Yes, we can see
Okay.
you, but the sound is a bit problematic.
How about now?
Yeah, it's fine.
Okay. Okay. So first, evolution. Now, evolution, as we know, is a defining characteristic when it comes to biological systems, right? But here we are going to talk about laboratory evolution, which is basically an evolutionary process that occurs in response to conditions imposed by the experimenter. So, and it can be monitored in real time by the experimenters. So, the typical workflow of a laboratory evolution includes the generation of the starting population, then to impose a selective pressure to get the evolved variants, and then analyze the mutations uh, to identify the causal mutations of the evolved strains. And next, it needs to be re-implemented in the production strains to validate the results of the increased production. But we are usually more used to using like random mutagenesis or targeted mutagenesis using PCR to generate libraries of selected genes in vitro, which can be rapid methods and very commonly used actually. But these methods sometimes tend to become very biased or have non-specific mutations, and which gives rise to the effective size of the library, which reduces the effective size of the library generated. So in contrast, laboratory evolution induces growth of the phenotypes with numerous intracellular pathways. So, uh, so laboratory evolution has been used successfully to engineer easily selectable phenotypes like utilization of nutrient source or tolerance to any inhibitory substrate or products for improving the production of chemicals in microbes directly. And the common methods of laboratory evolution includes batch culture, which is a very simple procedure which just requires regular dilution of fresh media. Then there is a chemostat, which includes a continuous supply of fresh media at a fixed rate. Then microfluidics, can which helps in analyzing the single cell at a single cells at a single cell level. Then there is emulsion culture, which basically forms vesicles of a mixture of oil, surfactant, and cells together. So it forms a large number of small population of cells in vesicles. So that is another way. And then there is mutation accumulation, which is again a very simple method, which introduces regular bottleneck into each replicate population. And also animal models are used in microbial cultures for some environments which are difficult to replicate in lab. So the microbial population is grown inside the animal model. So there are like these multiple methods which are used, but they have certain limitations like chemostat and turbidostat. Like chemostat continuously dilutes the culture at a fixed rate, but turbidostat can monitor its optical density and dilute it when the optical density exceeds a predetermined threshold value. But in spite of that, there is a great disadvantage to it, that it forms biofilm, which can which interferes with the function of the device and also dominates the dilution of the culture. So yeah. So next there is also microfluidic based continuous cultivation system, which is again, as I said, uh, which is very useful for analyzing cells at a single cell level, and also it prevents the formation of biofilms, but it has its own challenges, like it has a risk of losing information on slow growing but high production strains. And also, if there is any contamination, then it can get trapped in the structure of the channels in the microfluidic device. So then that really hampers with the design. Next, also there is droplet-based continuous cultivation system, which isolates slow growing but high producing samples. So using these methods, there are these methods are used to get a lot of to engineer a lot of microbes to increase metabolite production, which is like one of the foremost aims of metabolic engineering. So like in this case, Saccharomyces cerevisiae strain is engineered to produce an increased amount of carotenoids. So here they have used, so mostly they use selection pressures to manipulate these experiments. And here they have used hydrogen peroxide shocks at intervals. And as you can see that with each shock, it has spiked the production of beta carotene. And in the absence of hydrogen peroxide, the amount of beta carotene has drooped down. And this is visual representative of an increased amount of beta carotene against in the evolved population against the ancestor population. Similar engineerings have been done on Corynebacterium glutamicum strain, where biosensor, where they have used biosensor to produce an increased amount of L-valine, and they have so they have imposed artificial selection pressure here on they have imposed artificial selection pressure and measured the fluorescent output of the biosensor by using FACS, fluorescence activated cell sorting. And they have found that the L-alanine, which is a byproduct in the biosynthetic pathway of L-valine, the amount of alanine has reduced and valine has increased. So they have continuously bred that 10% of the high fluorescence producing populations and increase the production of L-valine. Again, increased production of tyrosine in E. coli strain by feedback regulated evolution of phenotype called FREP, which basically controls the mutation rate of genome based on the concentration of target metabolite. So the mutation rate affects the concentration of target metabolite over time, as a mutation that increases the metabolite production accumulates in the gene, and it creates a feedback loop which causes the mutation rate to decrease and the concentration of the metabolite to increase. So they have produced like 10 populations with increased tyrosine production. And this is a table of many such, a few actually, examples of such increased metabolite productions. And there are many more. So these are just a few of the examples. So in which laboratory evolution has been used to increase metabolite productions. And next, I would like to introduce one very important factor in laboratory evolution, which is fitness, which is actually an important factor if you consider evolution as a whole, because fitness basically, evolution actually depends on fitness to some extent, if you can put it that way, because fitness is basically a quantitative measure of the capacity of an organism to produce offsprings in the next generation. So the more fit the organism is, it is more likely to survive. So fitness assays are carried out to determine the degree of adaptation of a population after experimental evolution and to validate the fitness effect of any specific mutations. So it is done by again, there are various methods, like by employing markers and starting with a mix of strains, and fitness of these mutations can be monitored by monitoring the divergence of markers in time. And in fitness landscapes where fitness landscapes can be rugged or smooth. So suppose in condition 1 where the landscape is rugged, the strain A is more fit than strain B. But with a change in the condition, change in the environment, suppose the landscape becomes smooth, then strain A might become less fit than strain B. So in this way, like fitness can manipulate. Then there is diversity of effects of mutation that can be visualized as a distribution of fitness effects, where the deleterious mutations just they get eliminated from the population, and mostly a population comprises of having neutral or nearly neutral mutations, and very few beneficial mutations. So, and there are also barcode sequencing approaches which can be used to track lineages. Track lineages in microbial population. Now, with change in the environment, microbes adapt to the changed environment, and by some changes in their metabolic activities. So this is like a given thing, and from here there is a corollary that temporarily varying environmental conditions facilitates the establishment of complex metabolic traits. So if there is a starting environment with this red genotype being very fit in this environment, and with a sudden change in the target environment, the fitness peak might shift largely, which will render it to be unfit, render the genotype to be unfit, because it can't move like so much distance and get adapted and rise to the peak of fitness. But if you introduce an intermediate environment with just a slight shift of this fitness peak, then it might lie at the foothill of the fitness peak and slowly adapt itself and rise to a fitter version of itself. And then when you change the environment again to the same target environment, it will achieve its fitness peak. So here, changes in multiple changes in environment can thus facilitate the establishment of complex metabolic traits or new mutations, which can help in adaptation. So a few other terms which are really important to understand laboratory evolution are genetic variation and population size. So genetic variation determines the rate at which a population can adapt to the environment, to an environment, and effective population size basically detects the genetic drift in a population. The higher the genetic diversity, the lower will be the genetic drift, so lesser loss of genes. So this is a molar plot showing the frequency of different genotypes over time. If the rate at which new beneficial mutations appear is low, then one mutation will get fixed before a new mutation can take its place and before a new mutation can occur. But if the mutation supply rate is very high, and which can happen with a bigger population size, then multiple genotypes will compete in the population. So this allows for selection of more complex genotypes as more beneficial mutations can accumulate in the competing variants. Now we come to laboratory evolution. Laboratory evolution in antimicrobial resistance. Now, as we know, antimicrobial resistance is a global crisis, obviously, and antimicrobial resistance is a consequence of evolutionary processes. So, to combat antimicrobial resistance and also resistance to multiple drugs, evolution should also be taken into account. So, as I had explained before, evolution experiments can be used to measure fitness cost of mutations underlying antimicrobial resistance and mutation rate and probability of the evolution of resistance. So knowledge of the susceptibilities that evolve with multi-drug resistance could facilitate the targeted use of the drug combinations based on genotype of the clinical pathogenic strains. So here is a picture of a competitive fitness assay that where two strains with a marker are grown together and the cell ratio is measured after different periods of time. This is similar to the ones I have shown before. And next, this is what I am working on in our systems biology lab. And this is a where I have used laboratory evolution, and this is a major portion of my PhD thesis where we have used laboratory evolution to grow 1,000 generations of Saccharomyces cerevisiae populations in triplicates, and they show a high effective population size value, which means that they have a high genetic diversity. And I have tested their fitness values, and which shows that the relative fitness of the evolved generations and the relative fitness of the evolved generations is basically more than the ancestral strain, but amongst themselves they don't have much of a variation. So, which indicates that there is presence of neutral spontaneous mutations. So next, I'll be testing these populations against antifungal drugs to understand, to establish a quantitative relationship between genetic diversity and emergence of antimicrobial resistance, and whether the extent of resistance plateaus above a certain amount of diversity or continues to rise, or if there is any threshold value of diversity after which it gives rise to antimicrobial resistance. And I will also be testing these against environmental stressor to understand the contribution of neutral mutations on fitness across diverse environmental stressors. So yeah, this is what I'm doing after the labs open after this COVID. So these are my references. Now I would like to conclude yeah, so despite having some limitations, the laboratory evolution techniques have increased applications in advancing genome technology. And though natural fitness landscape can be more complex than the optimized laboratory setup, so it can be a bit difficult to get exact values maybe, but still it is it has been advanced enough, and it is provided a lot of insights into genetic and molecular basis of evolutionary changes in biology nowadays, and also in metabolic engineering. So hopefully we'll be seeing more of it. So these are my references. And I would like to acknowledge my supervisor and co-convener, Professor Ridhiman Dhar, and convener of this Indo-US symposium, Professor Amit Ghosh, our head of the department, Biotechnology, Professor R.K. Sen, and our Department of Biotechnology, IIT Kharagpur. Thank you for listening.
Thank you, Shreya. We have a question from the username Aram4244. So they're asking, how long does an ALE experiment take and which method is most commonly used? Batch, chemostat, microfluidics, etc.
Okay, so microfluidics actually is still I mean, people are still working on it. So I would like to say, I think the batch culture is the most common method of use, and it takes some time because it's evolution after all. So for an evolution to take place, it will need some time, right? Unless you are putting some extra selection pressure on it. Like I am working with neutral spontaneous mutations, so I don't have any particular selection pressure that I put on them to direct its evolution in a certain way. So for me, like I have generated 1000. If I can generate like 2000, 3000, it will be great for my study. Like there are this Lenski lab who have now like 70,000 generations of E. coli, and they are working on various aspects of that. So it takes a longer time than normal mutagenesis methods. And batch culture, this is like the easiest way you can go about it. And for industry grade, actually people can go for like chemostat and all.
Thank you, Shreya.
Yeah.
So now we'll conclude the students session and we will move on to the lecture series by the professors again. So I would request Dr. Amit Ghosh to kindly introduce our first scientist and dignitary. Sir, please.
Thank you, Manali. Let's start the talk with Dr. Srivastava from ICGEB. He is a group leader in systems biology for biofuel from ICGEB, and he earned his master degree from in chemical engineering from Indian Institute of Science, Bangalore, and thereafter he moved to USA, where he has done his PhD from Michigan State University. He won several awards during his graduate studies. Some of them are Sigma XI Award. And he then from MSU, he moved to for postdoctoral study in NIH, which is very popular, and especially in the area, he has done postdoctoral study in metabolism and won the award for research excellence in 2011. And then he moved to India in around 2012, where he started his own group. Now he is the group leader of metabolic engineering group for production of biofuel, and he has been really done well in recent times of discovering the different cyanobacteria, where he identified several cyanobacteria and genome sequenced it, and then have done the metabolic modeling, also 30C metabolic flux analysis, and then also doing the synthetic biology, metabolic engineering together. So in his lab, he developed all the techniques right from metabolic modeling, then 30C metabolic flux analysis. Also, he had developed software for doing 30C MFA. So he is a very knowledgeable person and very suitable for our conference. So he will be talking about his experience of handling different microorganisms in terms of modeling and then metabolic engineering, synthetic biology, and so on. So I hand it over to Dr. Srivastava for his talk. Welcome, Professor Srivastava, Dr. Srivastava.
All right. Just give me 1 minute. All right. Yeah. Good afternoon, everyone. And am I audible?
Yes.
Okay, great. So good afternoon, everyone. And first of all, I would like to thank Dr. Ghosh for the pleasant introduction and also for the opportunity to present some of our work here. And it's a great pleasure to be part of this meeting, actually, which has shown a lot of very insightful talks. And I look forward to hearing others as well. So today I'll be presenting primarily on the differences on different E. coli strains that we have been able to identify by 13C MFA, and with the with these analysis, a clear picture is emerging that obviously we should use different E. coli for different products. Now, generally, what has been done with E. coli has been restricted to a couple of strains, but obviously within the E. coli clade there is a quite variation in metabolism as well as genetic diversity. So with some of which I would cover today. I hope this would be useful and relevant to what is being discussed in this meeting. So before I go further, let me just briefly introduce about ICGEB from where I belong. So ICGEB stands for International Center for Genetic Engineering and Biotechnology. It is a organization in the United Nations system. So basically, it is an independent organization which does research. It has the funding for our research comes primarily from the member states. So we have about 60 plus member states which are member of the ICGB system. Now ICGB has actually three sets of what we call components, which are spread the headquartered in Trieste, Italy. Then we have a major center in New Delhi, India, and a recent one in Cape Town, and actually a very recent component has also opened in China. So ICGB is also expanding internationally. Our mandate is primarily to develop knowledge for the developing world and to be able to address the challenges and the problems that these developing nations face, and develop biotechnological solutions for those. So now some of you may be aware that we do a lot of research in ICGB from various publications, as well as from the news coverage that ICGB scientists get a lot. So not only do we do our own cutting edge scientific research in our laboratories and all the three components, but we also do support a lot of educational and research activities. And again, this information might be of use to a lot of students here. So we support advanced education through various long and short term fellowships for both PhD students and postdoctoral fellows. Now, I must clarify here is that these fellowships are for students to go outside their country of application. So if you want to go to another member country, say for example, Italy or South Africa or any other member countries, and you want to do part of your research there, ICGB may be able to sponsor your stay and research. But again, these fellowships are quite competitive, so you might need to apply for those. In addition, ICGB regularly organizes a lot of meetings, courses, and workshops. So like the one that you are just attending, if you go to ICGB website, in the section of meetings and courses, you'll find nowadays, because of COVID, a lot of conferences or workshops have been reduced. But generally, we used to have about around 10 workshops in different areas in ICGB. They used to be like where you come and stay with us for about 5 to 10 days or maybe more, and do not only theoretical work but also practical courses as well. So additionally, for young scientists who are slightly ahead in their career, we also have lots of competitive research grants for early scientists to return to their country of origin. Again, all of these are generally for member countries, and India is definitely a member country. So obviously the institute is involved in a lot of developing technologies for the developing nations, and a lot of other biosafety, bioethics, and other activities. So now from the introduction to ICGB, now if we talk about generally what are our unsustainable P's or problems, where P stands for problems, it's basically all of these start with letter P. So it's either pollution, environmental pollution, which is also partly caused by, in Indian term, it's called parali burning. So crop residue is burnt, and it is a major cause of pollution in all part of northern India, especially again in the months of November and all. You see Delhi has a very poor AQI, which is at least partly caused by these pollution through parali burning. Then the other problem we have as a country, and also a lot of other developing countries have, is their dependence on petrochemicals. Now, you may know that India imports about 83% of their petrochemical requirement. So our dependency on foreign exchange and the conditions that is imposed by the exporters, exporting countries is very much, and there is a strong impetus to develop our own technologies to supplement our energy demands. Then, of course, there is plastic and plastic waste, and significant, obviously, population is one. Pharmaceutical. There was a in the previous presentation, there was a talk about antimicrobial resistance and excessive use of pharmaceuticals and excessive use of pesticides. So all these P's are our problems, are our major challenges. Now, what I'll talk today has relevance to basically four of these. So basically, trying to address the pollution and the petrochemicals, plastics, and pesticides challenges of the world. Now, to address this, basically, the DBT was very kind, and it established a DBT ICGEB Center for Advanced Bioenergy Research. Our mandate is primarily to do identify and design novel cellulolytic enzymes, also to engineer microbes and algae for biofuels and chemical productions, as well as systems biology of bacteria and algae to aid in synthetic biology research. We have 5 PIs in the Bioenergy Center, led by Dr. Shamsi Asdani, and we have Dr. Shashi Kumar, who does algal engineering. Dr. Shams is famous for his cellulase work and also for his microbial engineering work. I take care of the systems biology component as well as a little bit of process development. And Dr. Pawan works in the area of algal omics, and Dr. Naseem Gaur works in the area of yeast metabolic engineering. So our areas of work is primarily could be divided into systems biology, genomics, synthetic biology, and fermentation technology. Obviously, all these components are needed to make a deliverable product in terms of which is viable commercially. Now, my group in particular actually works on a lot of different kinds of organisms. We work on yeast, we work on E. coli, fungus, as well as cyanobacteria. And Dr. Ghosh has told you about some work which we have done in case of cyanobacteria. Now, in all these organisms, now what we are trying to do in terms of systems biology is basically we have developed a lot of genome scale metabolic models of these organisms, which have helped to characterize these some of the non-standard organisms which are useful for especially for biofuel applications. Now, our group have also used a lot of methods to actually analyze this genome scale model and also to identify the targets as well as metabolic engineering strategies to improve the production of a particular metabolite of interest. Now, again, this can be any product that you want to make. Now, today what I'll talk about is a lot of our metabolic labeling studies, for example, 13C-MFA studies, and we have also done a couple of studies where just by analysis of the metabolic labeling patterns, we had analyzed or identified the quantitative contributions of the precursors to a particular product of interest. Of course, as I have mentioned, that we work in the area of process development and analysis, which includes scale-up of fungal enzyme and production. And again, in sync with what previous speaker has said, we are also looking at stress adaptation as a mechanism to improve productivity and the underlying molecular mechanism behind it. We have done some work in case of genetic engineering, where we have done expression of homologous and heterologous proteins in cyanobacterium. And this, so obviously some including traditional knock-in and knockout methods. Now, so our idea is that metabolic pathways are nature's solution to our unsustainable problems. So we have this KEGG metabolic pathway. Now we know that the metabolic product may act as either as additives. So for example, a lot of alcohol molecules can be additives directly to the petrol, and thus reduce the consumption, or direct substitutes for petroleum products or plastics. So some examples of biodegradable plastics are PHP, polylactic acid, and so on. Now, obviously, in addition to that, metabolites can also act as important precursors for synthesis of a variety of products. Actually, most of the petroleum derived products can be replaced directly or indirectly by uh, metabolic chassis components. But we also realize that metabolic engineering would be needed to overproduce the compounds of interest in organisms. So generally, you have been through this a lot, so I won't go into much detail of this. That obviously a lot of previous work has looked into pathway centric engineering. The problem is that obviously it does not always work. We think that the product should increase, it does not. And also it's difficult to quantitatively predict the cellular response to targeting a gene. So if I increase or delete a particular gene, how would my cell react? Now, an example of interest for a lot of you engineering students might be that, for example, the latest model of E. coli contains over 2,000 reactions. So, if we just want to delete 3 reactions in this number of reactions, the total more than 2 billion combinations are possible mathematically. And even for simpler network, the combination can be in hundreds and thousands. So obviously we need little bit of computational support to be able to identify accurate targets. So what we do is can be summed up as a bottom up systems metabolic engineering. So in the design, test, build and test cycle, we look very carefully at the design. The work typically starts with the basic metabolic measurements and genome sequence of a target organism, from which we generally reconstruct a curated genome scale metabolic model, and then we do the flux balance analysis of the general metabolism to see how the fluxes are distributed, what kind of engineering needs to be done. And in an ideal world, so for example, for a lot of E. coli work, we also want to do measure the intracellular flux distribution accurately through 13C metabolic flux analysis, and then to use this information back into our FBA to get a more accurate target prediction. So I'll briefly touch upon genome scale metabolic models also, because I think some previous presentations have touched into it, but definitely we must realize that for a lot of our non-standard organisms, which are not very common, but which are of significant interest for any biotechnological application, it will be very useful to have the genome scale metabolic model. As the name implies, it is basically a model which contains almost all the metabolic reactions in the cell, with the following qualities: that it is a model which has complete pathways. In a good genome scale model, there are GPR relationships. That means we know which reaction is conducted by which protein, and which is in turn coded by which gene or combination of genes. Additionally, it contains a biomass equation. Now, this itself is a very interesting thing to do, because generally uh, we do not take care that actually the biomass of the organism also varies based on the condition of growth and also the type of strain and so on. And you will see a lot of variation in the biomass composition of the organism based on the phase of growth. Additionally, a good genome scale model has mass balanced reactions with defined directionalities. And also for eukaryotes, the location of the reaction is also defined, as well as the required transporters. If we do it correctly, the model has the ability to simulate cellular growth and metabolism under a variety of different culture conditions. So obviously, it's not a simple thing to reconstruct a genome scale metabolic model. We begin with the genome sequence. It requires a good assembly and annotation, from which we get a draft reconstruction, from which basically now this refinement of draft is the most time consuming process, where basically we need to do a lot of quality assurances. So for example, that the reactions are charge and mass balances, a biomass equation is set up correctly, so that biomass equation primarily gives you the quantitative requirement for the growth of the cell. So how much protein it will take, what are the amino acid composition, how much lipid of 1 gram of biomass takes, and how much is the what is the lipid composition, and so on. Additionally, we also need to specify the ATP demand for growth and maintenance, and obviously the reaction localization. So once we do all this, we generally, this is an iterative process where we check the model through a technique called flux balance analysis, and we basically test for a lot of these criteria that A, it can make biomass through various precursors which are known to help in cell growth. Then B, it for all the known substrates and products, the model is, or at least most of them, the model is able to simulate the growth. Also for organisms for which known lethal knockouts are available, the model is able to simulate those. And in addition, the more extensive the model is, the better is the coverage for genome scale metabolic model. So a typical model may account for greater than 500 genes. So latest E. coli model has about 1400 genes and over 1000 reactions. And obviously it requires a substantial human effort, which is obviously not possible entirely with automated methods which are available. Now, once we do that, we can identify the unique pathways present, the growth and flux distribution under different conditions. So for example, for algae under different trophic conditions on different substrates and so on, and also identify the bottlenecks in the use of a particular substrate. So for example, lack of transport or a key enzyme, which can then be metabolically engineered. We can, as Dr. Ghosh has earlier presented, we can identify reactions which are essential for growth, simulate the effect of. Now here, this is what might be interesting for metabolic engineers, is we can do some basic simulations of what will be the effect of knockouts, both in terms of growth and product formations, or overexpression or introduction of heterologous pathways, and then which will help us in identifying genetic engineering strategies to improve particular target product formation. So I will not go into a lot of details of what models we have published, but these are all basically models which are relevant to bioenergy research. So we have made, the first model we made was for Geobacillus thermoglucosidaceus, which is an organism which is thermophile and can actually ferment both glucose and xylose. And this work was published in Journal of Biotechnology. Then we reconstructed for a marine alga. Again, this work was published in Algal Research. We recently, last year, we published for Methanococcus capsulatus, so basically which is a methanotroph. Now, this work was published in PeerJ and was the 5th largest viewed paper in this area. And then very recently, earlier this year, we published a model of a native marine cyanobacterium with very interesting properties. Again, I'm not going into these individual papers or individual models, because that's not the focus of the talk today. But definitely for any one of you who is working on similar organism, they are suggested to at least have a look of what kind of genome scale model it is and what it can do. Recently, we are working for an indigenous marine alga model reconstruction. So major biological insight is, of course, we basically identified the capability of the cells to produce various biochemicals of interest. For example, acids, alcohol under different conditions. Identified the bottleneck in the use of arabinose by Geobacillus. That is, it actually lagged a transporter. And in principle, if a transporter is added, it would be able to utilize arabinose. Then obviously for a lot of heterologous and homologous compounds, what are the metabolic engineering targets? What are the theoretical yields that can be got? And also we predicted the formation of hydrogen by Nanochloropsis gaditana, and actually a lot of Nanochloropsis some strains at least have been shown previously to produce hydrogen. Similarly, we also predicted the activity of alternate pathways in these organisms. So if there are more than one way of producing a particular molecule, then these analysis of these genome scale model will tell us whether pathway A is used or B is used primarily. So today I'll talk about 13C-MFA. So previous talks have discussed it in a little bit of detail. So basically, we start with labeling experiments. Now the aim generally is, at least for steady state metabolic flux analysis, is to reach isotopic and metabolic flux, steady states. So now from the cell, basically we look into the metabolic label incorporation into different amino acids. Now, for example, let's say for valine, we can say what are the different enrichments at different positions, whether all the three out of the four carbons are enriched, or and so on, and how much is the relative enrichments. And we can also identify, we need to identify the external rates. So for example, what is the rate of growth, glucose intake, and metabolite production, and so on. All this then feeds into a metabolic model. Now, obviously, for an organism like E. coli, it is well established. For a newer organism, it needs to be established, which is then put into a 13C MFA software. Now, in this case, in the work that I'll present, we have used a software which we have only designed. And then obviously you need to ensure that whatever is model is simulated fits the data generated well. And ultimately, if everything goes well, you get a flux result, which basically defines the metabolic map of the organism. Now, in terms of 13C MFA, we have actually created a open source software which is a Python based for MFA calculation and visualization of results. Now, a lot of other software that are popular are based on MATLAB. Now, for IIT students, MATLAB is freely accessible, but for others, actually, it's a fairly costly software. So this FluxPit is actually is a option which is entirely Python based and completely free to use. We have published this work in PRJ, where we have actually shown that the calculated flux distribution is identical to what is predicted by MATLAB based software. So obviously it requires input of model as well as flux values. And then we also need basically atom transition, reaction atom transition information as well, and the MID data, what is the labeling data in various metabolites, from which basically a lot of internal calculations occur to reduce the error between the calculated and the measured MIDs, and based on these the flux distribution is calculated. So again, you guys are welcome to look at this software, and if any one of you is interested, we can also talk further on that. So I'll talk about E. coli here. E. coli is actually a very useful organism. There is no need for me to tell anyone this, but it is also the most studied and characterized organism and can grow in minimal media and consume both glucose and xylose. Now this property is very useful for biofuel research, because a lot of because primarily the lignocellulosic hydrolysate that is prepared contains these two sugars. Xylose is not utilized traditionally by Saccharomyces cerevisiae, and of course a lot of engineering effort have been done to engineer S. cerevisiae for xylose metabolism, but E. coli can naturally utilize xylose. The other advantage it has is that several genome scale metabolic models are available, and also 13C MFA is fairly well established. Additionally, the important here is that tools of engineering are also well established. So in this work, we have basically used 4 different E. coli strains. E. coli MG1655, which is a very popular strain, which is a derivative of K12 strain, very popular in metabolic engineering. And actually, the very popular DH5 alpha and DH10 alpha, which are used in a lot of cloning work, are in fact derived from this E. coli strain. Then we have E. coli B, which is the parent strain for BL21D3, which is mainly used in gene expression studies and also has been engineered for efficient xylose to ethanol conversion by Dr. Sham's group. Then we have E. coli C, which is actually morphologically distinct from other E. coli strains, and it can also form robust biofilms. The other last strain that I'll talk about is the E. coli W. This is also a very interesting strain because it is the only E. coli strain which can also utilize sucrose. It has very less acetic acid production. Some of these is also used in cloning purposes. The other reason of choosing these E. coli strains is that these are all biosafety level 1 strains. Now, whenever we talk about a lot of metabolic engineering research, uh, there is very less discussion of the biosafety aspects of the organism, which is also very critical to take care, because uh, the higher the biosafety level, which means that the organism is not safe for human and may cause diseases, the higher becomes the burden when we scale up things. So in this work, basically we did anaerobic culture because for a lot of molecules which are biofuel related, anaerobic conditions are employed. And this was done in serum bottles. So this medium was actually bubbled very well with nitrogen to ensure that there is very minimum dissolved oxygen, and then the culture was grown. So of course, after growth, the medium became turbid. So here in this work, we have used 2.5% glucose, which is uniformly labeled glucose, as well as labeled at first 13 carbon at 80 to 20 ratio. And for this work, we have used multiple bottles, one for each time. The pH was adjusted throughout the hourly through injection of a base. And also the extracellular fluxes here were measured using HPLC, and metabolic labeling of cellular proteins was measured through a GCMS. As mentioned, the fluxes were calculated using FluxBid. So, I mean, this figure tells you how different just on glucose these different strains are. So, basically, if you will see that E. coli C, which is known to be different than other strains, produced much higher levels of succinate. Even in case of other products, there is a significant difference that is seen in terms of the strain performances. Some of them made certain acids more compared to other, and so on. Now, in terms of the yield, uh, obviously the biomass yields were slightly varying, like which is shown in the blue at the base, but they are within the range what has been reported for anaerobic cultivations. And obviously, E. coli C had a very high succinate yield. So now, if you would look here, you will see that these are 3 different E. coli strains. They have almost double the difference in terms of their growth rate. So E. coli C had almost the half the growth rate of E. coli B under similar conditions, under anaerobic conditions. The same is with the glucose intake flux, which is about 50% higher in E. coli B compared to MG1655. And the same things, you will see a lot of significant differences in both in acid formation as well as ethanol formation. And again, you will see succinate flux is so high. It's like 10 times for E. coli B for E. coli C. So these strains are very, very different metabolically. Now, in terms of the metabolic flux analysis, when this was done based on all this data, it told us that, for example, E. coli MG1655 has very poor or relatively poor pentose phosphate pathway activity compared to E. coli B, and so again about a thrice, 3 times higher PPP activity in E. coli B compared to Mg1655. Also, obviously, the fluxes to products were higher in E. coli B compared to Mg1655. The other interesting thing that we have observed was that TCA cycle is not completely running. In fact, it is running half, and there is very little flux from here to here. And in certain cases, for example, Mg1655, again, it is running about until here, AKG, which is used in biomass formation. And then there is another half that is running to produce succinate. So this was very interesting insight that was revealed by this analysis. Then also for E. coli C, what we have found was it has a fairly robust pentose phosphate flux, and as well as obviously here also the TCA cycle was running in two separate fashions. So one way is like here to produce succinate, the other is coming from pyruvate to oxalate to formation of succinate. So obviously, what we can summarize is basically what we have seen consistently in all the three strains is that under anaerobic condition, ED pathway is not active. Also, the TCA cycle does not run completely in anaerobic conditions, and the PPV flux in E. coli B as well as E. coli C is about 3 times that in Mg1655. Now, we wanted to see whether this can also translate to response to xylose fermentation capabilities. So we also grew these 4 strains, although I have not shown the 13C MFA of E. coli W. This was the strain that we had recently procured, so we are working on the 13C MFA of this strain as well. But we are seeing again a huge difference both in terms of growth as well as product formation, also on xylose. So for example, both Mg1655 and E. coli B have fairly comparable growth rate, but E. coli C has a very poor growth rate uh, in xylose. And again, if we look at, so starting OD in this case was about 0.03. So about 10 hours on xylose, which is again, xylose or anyway growth is poor. The OD of E. coli C was just zero 0.8, while for these Mg1655 and E. coli B, it was about 1.4, but E. coli W had an OD of 2 and the growth rate of about 0.62, which was the highest. Sorry. Also, in terms of product formation, we have seen significant differences. So E. coli C did not consume xylose completely even after actually 16 hours. The same was with the E. coli Mg1655. The consumption was less, and as you will see, there was again W was about 50% higher than Mg1655 in terms of xylose fermentation capability, and this was seen throughout. Mg1655 and E. coli B were fairly comparable. Again, E. coli B was slightly better in terms of general fermentation characteristics. Uh, but obviously, E. coli W was significantly better than both of these. Now, so from what I have presented, I hope I was able to convey that basically metabolically there is a very significant difference in growth and fermentation of different E. coli strains. While E. coli B may be very attractive organism for glucose metabolism, and E. coli C may be useful for protein production based on its active TCA cycle and pentose phosphate pathway activity. It is very poor for xylose fermentation, for which actually E. coli W could be a very significant strain. Also, interestingly, we see there is no correlation between PPP activity determined with 13C glucose labeling and xylose fermentation ability of the strains. So, which is also interesting that once the carbon source is changed, the fermentation performance is also very slightly different. So basically, all this information can actually now go back into the genome scale metabolic models of these E. coli strains, where we can now restrict um, the flux ranges and get a much more accurate genome scale level flux distributions, which can then feed into identifying metabolic engineering strategies for different products. What this can lead to is basically we can identify which particular strain is good for which particular. So for example, if I want to make a product X from glucose, which of the strains might be slightly better, and if I want to make product Y from xylose, which of the strains might be better. Though obviously from the face of it, it looks like E. coli B and W might be the answer for these two questions, but interest the significant differences in metabolism are indeed interesting. So with this, I'd like to acknowledge my team, which is working very hard to do various experiments as well as modeling work. Trunil is the one who has done a lot of this work. In fact, he's leading the 13 CMFA work that is being done. And incidentally, he's also a product of IIT Kharagpur. I'd like to thank Department of Biotechnology for the constant encouragement that we get in terms of funding, and also all the national and international collaborators on various other genome scale metabolic model work for their help and support. So thank you everyone for your attention. Thanks.
Thank you, Dr. Srivastava, for a wonderful talk, insightful talk. Now the session is open to question. So we have a few questions from the audience. The first question is about MFA.
Yeah.
In MFA only for estimating fluxes in central carbon, is it only used for central carbon metabolism, or it can be used for entire metabolic network? This is the first question.
Is MFA only used for estimating flux in central carbon? Can it? Of course, as Professor Amit has recently presented, the next level in 13C MFA is actually genome scale 13C MFA, which is basically for slightly higher level network. But currently, traditionally, again here, what we also have to understand is basically the resolution of the fluxes or the flux confidence intervals that we can measure. Now, what I have not presented here for the sake of simplicity is also the confidence. Whatever value I am telling you is basically the average value. I also want to be sure that what is the range also should be tight, so that what I am saying is close enough. Now we have to take care of that. Now, within that caveat, obviously newer methods are being developed where in theory we can do genome scale level 13 CMFA, but that is recent development.
Okay, the next question from the audience. Dr. Anu, you want to ask? So the second question, can you use non-glucose carbon source for 13C-MFA, or just you can use glucose only?
So of course, you can use the non-glucose carbon sources, because ultimately what you are looking for is incorporation, and as long as the metabolic network is well defined, including the atom transition. So for example, basically if you know what are the pathways, and also that here another information that is needed is what carbon of the substrate goes to what carbon of the subsequent metabolite, and so on. If that information is available, in principle, this can also be applied for non-glucose substrates. As a matter of fact, xylose based 13C-MFA was done some time ago, again for E. coli.
Okay. Okay, from my experience, I actually did 13C-MFA in galactose. So the only problem is that when you use glucose, it is cheaper, and if you use galactose, it is 4 times costly. So you have to be yeah, economical in when you do the experiment, because in the yeast we generally we use yeast for 30 CF, and the yeast the galactose promoters are very strong compared to glucose promoters. So, but you have to. That's why we
Correct.
prefer to change the promoter rather than putting extra burden on the experiment, like buying so
Correct.
expensive raw material. So you
Absolutely.
can do any raw, I mean, a substrate you can use. You can just buy. It will be super expensive because glucose is most widely used. So that's why when
Yeah.
anything which is used extensively, that cost of production comes down. And anything which is not used by many people, that means only it will be made for you, and that's why the cost of production goes very high. Okay.
Right, right. For example, again, here we have also used 113C carbon. So uniformly labeled carbon is quite cheaper. 113C is
Hmm.
costlier. Now again, based on the resolution at the or the confidence interval that you want at particular pathways, you might need to tailor your uh, label that you are using, or a combination of that, which again can be costly. As well as, for example, xylose, labeled xylose, if you want to use both uniformly labeled as well as specific labeled, it can be prohibitively costly actually. So here, that is the major determining factor to certain extent.
Okay, I have one question. Like you have mentioned about strain B, C, D, and W. Is there this is an engineer strain or just wild type strain?
No, these are all wild type strains. These
Acha.
are all wild type strains. Actually, if you would read the basically the uh, way these strains have been passed on from one lab to another, and in certain cases the background is also not clear, but this is very well clear that these are separate strains based on various genome level analysis as well as metabolic characteristics. But very actually no other study that I know of has looked into these comparative metabolic performances of these
Mhm.
strains.
Yeah, and in terms of glucose substrate consumption, also same for all these strains. It can consume xylose or
But then there are certain interesting differences. So for example, E. coli W can utilize sucrose fairly okay, but other strain cannot. So there is very interesting differences between these strains as well.
okay, one more question is that you have done a lot of metabolic modeling in photosynthetic microorganism, and especially for cyanobacteria and algae. So, what are
Right.
the challenges you face, like when you build the model, especially when you
So
take
again, yeah, that's a good question actually, and that's something which we face on a daily basis. First of all, for any non-model organism, especially just get, I mean, just the surety of the pathway becomes a little bit questionable. So we have to be very careful with what has been established. So compare the genome sequences fairly well to ensure that it is there. The second thing is the especially for cyanobacteria and all, the flux values are very poorly known, and there are only a couple of 13C metabolic flux studies in certain organisms. So to see a comparative distribution with what is known is very different, because again, I will give you example of the cyanobacteria that we are working on. Its biomass composition is entirely different than what for other Synechococcus strains. So obviously the internal flux distribution will be different. So those are certain challenges, and then also knockout information is not available at that scale. So the quality assurance there becomes slightly lower because all this background information is lacking.
so you start with a draft model, like you use model seed, or you study your own draft model in the sense.
So generally we start with our own draft model.
Okay.
For only I think one or two cases we have done with model seed, but then it becomes little bit more complicated. So when we have to actually correct a model, a genome scale model, and go back and check whether this reaction occurs or not, that becomes even more challenging to address. So I think there is one more question. If product is growth associated,
Yeah.
should we consider both
Ah,
biomass and? maximization. So if it is growth associated, obviously biomass maximization should do it because it is growth associated. So ideally, if the growth increases, your product formation also increases. So again, ethanol is a typical example.
Just we missed few word connectivity, I hope. Hello, is it audible?
Yeah, you are.
Yeah. Okay. Okay. Thank you, Dr. Srivastava, for a wonderful talk. Really nice work you are doing in ICGEB in terms of modeling and experiment combining these two things together, especially in Indian point of view, is very rare. Only ICGEB has those facility. Like you have extraordinary facility in terms of by measuring the metabolite. You have extraordinary facility and state of the art facilities there. Really appreciate your work, and we keep on. We'll have the physical meeting also in near future. Basically,
Right,
this meeting.
right. No, definitely we are blessed. It does require.
Especially Professor Tang told today morning that definitely going to meet in summer if possible, situation improves, and then we can have a meeting offline. And, can
Yeah,
have a group of metabolic engineer from India who can actually guide young minds who are actually will be leading science in
yeah,
future.
that would be good. That would be very good actually.
Lot of
Yeah, yeah,
interest,
absolutely.
a lot of interest from this workshop. I have seen like several hundred of people have registered for the workshop, and
No, no, I think yeah, it was a good, and the lectures were also fairly good.
yeah,
Talks were
yeah.
pretty good.
Okay, thank you. We'll meet you soon in person, and we'll discuss more. And thank you for your valuable time, and we really enjoy your talk. Thank you. Bye.
All right. Thank you, Dr. Ghosh. Thanks. Bye.
So our next talk is given by Dr. Anu Raghunathan from NCL Pune, and she did her PhD from IIT Bombay, and then she moved from India to US in University of Florida, where she has done her first postdoctoral fellow in microbiology, and then after that she moved to UCSD, University of California, San Diego, with Bernard Paulson. So, as you know, the Bernard Paulson is basically the founding member of. He is the founding member of systems biology, and most of the method we use today is actually developed by him. And it will be great honor to actually listen to Dr. Anu Raghunathan for. And then after that postdoc, she moved to as a faculty in my in US. She was faculty for Mount Sinai Medical School, where she was there for several years, and then moved to India and become a senior scientist in NCL Pune. She has been working on metabolic engineering for production of plastic, and then also working in cancer cell line and so on. So it will be great honor and great it will be great privilege for us to listen from Dr. Anu Raghunathan, and I have a great interaction today. So I invite Dr. Anu to actually start presenting. Thank you.
Can you see my screen?
Yeah, no, I mean, screen is Screen is not visible.
You're unable.
You have to share the screen.
Oh. Oh, it's Can you see my screen now?
Right. Great.
Okay. All right. And can you just see my screen now, or can you also see my, I put it on presenter tools. Can you see presenter tools or only my slides? Hello?
We are seeing both your presentation slide
You
and
are
as
seeing
well.
both, is it?
Yeah.
I see, I see. All right. So I'll just put it on slideshow then. Okay. Hello, everyone. And at the beginning, I'd like to thank Amit Ghosh, Dr. Amit Ghosh, for inviting me to share some of my thoughts in this workshop for metabolic engineering. And it is an equal honor for me to be a part of it. And I hope I can keep up to Amit's expectations. So my title today for my talk is Engineering Pathways in E. coli using Systems and Synthetic Biology Techniques. And what I will walk you through in this talk is through the paradigm of systems biology and how one can use it to engineer pathways in E. coli or any organism. So as I begin, I'd like to bring your attention to some of Francis Crick's thoughts on scientific discovery that he wrote in his book, What Mad Pursuit. And one of the things he says is that almost all aspects of life are engineered at the molecular level. And without understanding molecules, we can only have a very sketchy understanding of life itself. So what I believe Francis Crick was trying to point out over here is that it's not enough to just have the delineation of all the molecular hierarchies in the cell, that is the DNA, the RNA, the protein, and the metabolites, but it is necessary to understand their interaction to be able to predict anything. He also quotes in his book an agenda for mathematical modelers in biology. So he says that the job of theorists is to suggest new experiments, and that a good theory makes not only good predictions, but surprising predictions that turn out to be true. Now, this fact, these molecular hierarchies in the cell have been talked about right from the first talk today, and I will try to show you how their interaction really is helpful in designing strains to produce a product of interest. So with this background, I would like to give you an outline for my talk, which would include technology, computation, and biology. And essentially will walk you through different steps that tell you how can you design a rational strain for PHA, that is polyhydroxyalkanoates, which is bioplastic. So we'll go through different techniques that exist in technology that is related to DNA synthesis, DNA optimization. We'll move on to genome, some aspects of genome engineering and the need for it. We will look at how we use codon adaptive indices and transcriptional initiation rates to sort of guide synthetic biology principles, and how do you use constraint-based reconstruction and analysis or flux balance analysis, which a lot of speakers today have alluded to, in order to end up with one iteration of the story of how one can make polyhydroxyalkanoates. That is via the systems biology paradigm, which is essentially component enumeration, reconstruction, modeling and simulation, hypothesis generation, and biological discovery. So metabolic engineering in the broadest sense, as we all know here who are attending this workshop, is the overproduction of a desired product of interest in a microbial strain. So metabolic engineering essentially views the cell as a chemical factory. And just as the chemical factory has different component classes like unit operations like mixers and reactors, connectors like pipes and tanks, and controllers like instrumentation and valves, Analogous to that, a microbial cell has organelles, proteins, genes, and regulators that need to be fine-tuned to achieve the level of sophistication that exists in a chemical plant. So in order to do that, we need to reconstruct the system, because the whole metabolic pathways that exist in a cell are very extensive, as shown by many speakers in previous talks. So how do you reconstruct a system? So there are multiple ways of reconstructing a system, and the kind of annotation our lab does, or reconstruction our lab does, is two-dimensional annotation of genomes using the matrix formalism. So we start with the primary annotation of the genome, and we delineate open reading frames. We understand what are the open reading frames. And say, for example, this green strip here, the green, red, and the orange represent three different open reading frames. So the green one can represent DNA, which codes for a regulatory protein that would bind to the DNA again, to the genomic DNA, and either repress or overexpress that particular gene. We also have this red open reading frame here, coding for a protein product like this triangle, and this orange open reading frame coding for a square, that come together to form a protein complex that can then catalyze a reaction like an isomerization reaction going from A to B. Now, there are many ways you can represent these in a reconstruction. But how we represent it is we look at all the links. And if you see here, we have represented a matrix. We represent components as rows, and we represent the links as columns. So if you look at the first interaction, the DNA protein interaction, you see that there's a circle, the DNA, another piece of DNA that that particular protein binds to, the purple line, and then the complex. So we basically assign elements of stoichiometry in these columns of the matrix based on based on the formation of this DNA protein complex. So as you can see here, the next reaction is two proteins coming together to form a protein complex. So you have anything that is consumed, as you all know, in stoichiometry is given a negative sign. And the stoichiometry here is 1 is 1. So they both come together to form a protein complex. And then in the isomerization reaction catalyzed by this protein complex, you have A going to B. So A gets a value of -1 and B gets a value of 1. So you can see that just with 3 open reading frames here, we end up with almost a matrix of 3 plus 3 plus 2. That's 8 by 3. So the first small part of the matrix here represents regulatory interactions. The second part here represents direct gene products. And the third is an effect on the downstream compounds. So this whole process is called the reconstruction of a system. And this process is, this matrix that we get is called the two-dimensional annotation of this system. What is the advantage of having such matrices represent genomes is that you then have the tools of linear algebra and convex analysis that you can use to sort of manipulate and compute cell phenotype. So as an example, I have here a set of reactions that represent glycolysis. I have the stoichiometric matrix for glycolysis represent here at the bottom left. And here I have the genes that code for, some of the genes that code for the proteins that are involved in glycolysis. So the concept of stoichiometric network reconstruction and metabolic network reconstruction essentially ties up biochemistry, mathematics, and genetics. For example, here, you can see that the enolase reaction is 2PG going to H2O plus PP. Now, enolase is this reaction here. Oops. So the second last column in the matrix. So each reaction is represented as a column, and the stoichiometric elements are represented against each element or each compound that participates in that reaction. So 2Pg would be consumed, so it gets a negative sign, and it's only one molecule of that, so it becomes -1, and gives an H2O and a PEP, which both get +1 as their coefficients. So you can see that such reaction vectors. So each of these columns actually represent the chemical reaction vector for this particular chemical reaction. And because it takes place inside a cell, we call it a biochemical reaction vector. Um, the right-hand side depiction essentially is of the gene B2779 that encodes the enolase protein that eventually forms the enzyme that catalyzes the reaction that we are all aware of. So here it is a single gene protein reaction relationship that is elucidated over here. So we use Boolean relationships to represent the genetics behind each reaction that exists in this stoichiometric matrix. So when you look at a model, like for example, when you look at a MFA model that people have been talking about in many talks today, what you actually look at is this outermost layer. This is the outermost interface that you look at, the stoichiometric matrix. But what exists behind it, it is what allows one to understand, what gene can you knock out that would improve your product? How do multiple genetic interactions change the side as a toxic side product or energy formation, and so on and so forth. So these gene-protein reaction relationships are very important, and I think even Amit in his talk alluded to them. So you can essentially depict isozymes, you can depict multimeric proteins, you can depict multi-protein complexes with multiple types of and or relationships that form the basis of Boolean algebra. And that way you can actually understand the exact impact of, say, if there's a mutation in SDHA, how would that impact the function of succinate dehydrogenase, for example? And especially in the context of metabolic engineering, transport of substrates is very critical to the conversion and the yields to products of interest. So as you can see here, this is just the PTS glucose system in E. coli. And I can't even read out the names of the genes here or the proteins involved in the system. But as you can see, it's really complex. There are more than 14 proteins involved here. So this essentially means that we need, in order to understand the pleiotropic effects of metabolism, we need to develop a computational model. And so to get to the grand challenge of metabolic engineering, which is to create a cellular system that is both efficient and economical, and at the same time operates at stoichiometric yields and rates, we definitely need to create a metabolic reconstruction and then eventually a model that can help us compute cell phenotype, understand redundancies, understand energy requirements, cofactor requirements, and any toxic side products that can be formed that would reduce the biomass. Because as you all know, the cell has evolved has evolutionarily, evolved to have the function of growth. So the evolutionary principle is survival of the fittest. And so it is not to satisfy our bioengineering objective. And so in that context, we need to sort of challenge evolution. And we need to make sure that in order to create a cell factory, that the cell not only makes biomass and is happy, but it can also produce the product of interest for us. And hence, to design the strain and increase the productivity of any compound of interest, we need to maintain simultaneously a redox balance We need to prevent side products, as I just mentioned. And so this requires the integration of both the reductionist understanding of pathways, that is the component enumeration, and the integrative knowledge of how these pathways are networked. And this, as you can understand, the complexity of metabolism warrants a computational model. And as C.A. Hutchinson mentioned, I think in 1972, the ultimate test of understanding a simple cell, more than being able to build one, would be to build a computer model of the cell, because that really requires understanding at a deeper level. And so this is how one can move from biological network models to whole cell simulators, and hopefully one day cell factories, willy-nilly. So once you have a network reconstruction, you can then, a network reconstruction has a lot of detail that is associated with it. You need to know where these reactions take place, where these components are present. In case of E. coli, is it present in the periplasm or the cytoplasm, or is it an extracellular metabolite? In the case of models like yeast, which is what a lot of people have talked about today, you also need to know the compartmentalization. So there are almost 9 compartments in yeast. And so you need to understand whether a particular metabolite or reaction is present or takes place in the Golgi apparatus or in the endoplasmic reticulum, and how the transport between these two organelles takes place. Stoichiometry, as I just mentioned, is very critical. It forms the basis of this metabolic model. And if you have incorrect stoichiometry, you are probably going to have incorrect results. You need to make sure that all your reactions are consistent as well. The cofactors, the choice of cofactors is also very critical to being able to balance cofactors when you are making a product of interest. And the use of the right cofactors is also essential in a reconstruction. Reversibility of reactions or thermodynamic considerations are also critical. So is the localization, and any biomass or ATP requirements or regulation would eventually help you in setting up constraints, which I will talk about later. So once you have a reconstruction in place, and this process actually takes about 2 years if you start from scratch. I know one of the speakers alluded to the existence of a lot of automated network reconstruction engines like Model Seed, etc. But even if you use those engines to create a model, a base model, it is very essential for the person who's going to use that model or that network to go and curate each reaction at a time. So it approximately takes one person about 2 years to make sure that they have a good model. And that is for a model that has enough literature. So for models like, non-model models, non-model metabolic reconstructions, like I think the speaker was alluding to, it takes even much longer because there's not enough literature that is present. E. coli, which is probably the workhorse of biotechnology and everyone is familiar with, there's only about 50% or 55% of the genome that we actually understand today. So then you can probably extrapolate what the situation might be for other organisms that are not as popularly used in research like E. coli. So the phase 1 is to reconstruct, to create a reconstruction. The second phase is to create a metabolic model. And the third phase is actually to run testing and validation and simulations. And I will talk about this process a little later. After I allude to the compound we are looking at in our lab. So in my lab, we look at multiple compounds of interest. One big interest of ours is drugs and pharmaceuticals. So we started off with a very small molecule, which is the condensation product of 2 tryptophan molecules. Tryptophan, which is an aromatic amino acid, when it condenses, it forms a violet-colored molecule called beerlacin. So we developed some prototypes to make that particular product. I was involved in another Gates project to make kanamycin. And currently, we've been awarded a very big proposal from CSIR under the vertical of import substitution to make penicillin, which is, although it is not used by the medical profession these days as an antibiotic, it's an API that can make a lot of semi-synthetic penicillins. And if any of the students here are interested, we have some openings in the project. So please feel free to write to me anytime, and we can discuss potential employment over there. But today what I will talk about is another interest of my lab, that is polyhydroxyalkanoates or bioplastics. Now, why polyhydroxyalkanoates? Drugs are very obvious to understand, right? So PHA is an alternate to current plastic polymers like PET, polyethylene terephthalate. PHAs are biodegradable, biocompostable, biocompatible polymers, and the availability of monomers and the cost of them is quite prohibitive. And so it is essential for us to create a sustainable process. And hence we are interested in developing a cell factory design, a rational strain design first, and then a cell factory design for PHA. So what we are trying to do in my lab is, try to employ different areas or state-of-the-art methods in synthetic biology, in metabolic engineering, in systems biology, and in evolutionary engineering. So we can convert sugars, or in the future, renewable biomass like cellulosic biomass, like the previous speaker alluded to make chemicals, drugs, and bioplastics. Now, these are some of the pathways required for PHA production. So you essentially start with acetyl coenzyme A, which is the byproduct of glycolysis, and that siphons, that sort of tags into the TCA cycle. Now, acetyl coenzyme A, instead of going into the TCA cycle, if a part of it is siphoned off, it can be converted to acetoacetyl coenzyme A, which can then, along with the products of the fatty acid beta oxidation or fatty acid de novo synthesis, in the presence of the enzyme PHAB, it can produce a 3-hydroxyacyl coenzyme A, which is essentially the monomer for a polymerization enzyme, the PHA polymerase, to make any polyhydroxyalkanone. So essentially, if you want to make a PHA, you need to have lots of acetyl coenzyme A. And I think a lot of speakers have made my task very easy because, a lot of them needed acetyl coenzyme A to make their products of interest as well. Now, what we have done is, so this particular pathway is present in Ralstonia, Eutropha, and a lot of other microbes which are a bit messy to grow. So what we decided is we would harness the, the favorite workhorse of biotechnology, that is E. coli, and put this pathway into E. coli. But then this would be another, so we had a lot of experience with the vialicin pathway, which is actually a 5-gene pathway from tryptophan to vialicin. And we saw that it was very difficult to actually engineer 5 genes into E. coli. So we wanted to see, are there native pathways in E. coli? Can we exploit the redundant metabolism, the native metabolism that exists already in E. coli, and transplant a minimal set of genes from an organism that actually is known to produce or natively produces PHA, so that we can sort of save ourselves a little bit of work for genome engineering. So E. coli produces 3-hydroxybutyryl coenzyme A. And so we decided that as a prototype, let's try to make let's try to make polyhydroxybutyrate, which is essentially the product that comes out of a polymerization reaction if the monomer is in high concentration. And the monomer here I'm talking about is 3-hydroxybutyryl coenzyme A. So we were not just interested in creating PHB, because PHB has been made by multiple groups around the world. And we were actually, what we wanted to do is we wanted to go one step down and make the smallest polyester that is feasible, and that is polylactic acid. So although we don't call it lactanoate, polyhydroxylactanoate, if you have lactoyl coenzyme A as a monomer, you can actually polymerize it to polylactic acid. And so some of the genes that are required for small molecular weight, small chain polyhydroxyalkanoates, we decided to go and prospect for them in different organisms. So what we did is we found that there was a propionate coenzyme A transferase, which essentially transfers coenzyme A from both propionic acid as well as acetic acid to lactoyl to make lactoyl coenzyme A, and so you can use lactate as a substrate here. And we thought this might be a good a gene or enzyme to have in E. coli if we wanted to make small fatty acid chain polyhydroxyalkanoates. We also chose the polyhydroxyalkanoate synthase from Chromobacterium violation because the PHAC that exists in Chromobacterium polymerizes small length hydroxyalkanoates. And I apologize for the typographical error here. It should be alkanoate and not alkanate. Right? So, so it's a type 2, PHAC is a type 2 synthase for low molecular weight monomers. And so we decided to go and source these genes from these organisms, the Acinetobacter pasteurianus for PCT and Chromobacterium violation for PHAC. Then we decided to use some of the basic principles of synthetic biology why use synthetic biology, right? I mean, people have been doing recombinant techniques for many decades now. Is it just that, we found that term fancy and we wanted to use it? No. So as I mentioned in the previous slide here, when we went bioprospecting for genes and enzymes that we could add to E. coli, we found that the genes of interest were present in 2 different organisms. So one of the reasons polylactic acid is not made in nature is because there are no microorganisms that exist with the capacity to make lactoyl coenzyme A and also polymerize the lactoyl coenzyme A to PLA in the same microorganism. So we really had to search a lot in literature and prospect to identify these 2 organisms. And so if you have if you have genes from 2 different organisms and you want them to express in E. coli, then the DNA is not going to be optimal for transcription and translation in E. coli. And so there's going to be a lot of multi-organism codon bias, and the specific codon frequency is not going to be optimal, and neither is the codon adaptive index. And we also saw that the GC content was varying between all these 3 organisms. And another reason to optimize genes using the synthetic biology approach is that you want to eliminate restriction sites, and you want to remove cis-acting elements or any tandem repeats because you want to prevent host nuclease cleavage. And that is why we decided to go with the gene optimization process, which essentially optimizes the codon to a CAI greater than 0.9 in relation to E. coli, which allows for faster translation to higher CAI. We kept the GC content between 40 to 60%, which allows the synthesis process. So this is actually more of a manufacturing problem. The chemical synthesis of the DNA is easier when we restrict this. And of course, it has a lot of other implications in the organism. We wanted to eliminate the risk of DNA damage by host nucleases. So we removed a lot of the restriction sites and kept only a few. And then we removed the tandem and inverted repeats so that, we don't get any, any further problems when we integrate these genes into the E. coli genome. Yeah, so for the PCT gene, we did that. So as you can see, we've changed the codon adaptive index to 0.9. the effective codon frequency is about 79, I think 74. And the GC content was adjusted to 51. It was originally 35. And the CI was originally 0.57 and it was made to 0.9, and we removed a lot of the cis-acting elements. And so we presume that the PCT now is modified for E. coli and will have optimal transcription and translation in E. coli. We did a similar process for PHAC, and we ended up with, we were only able to modify it to get a CAI of about 0.88 in E. coli. The codon frequency also was not that high. It was about 69, but this was the best we could do in order to, maintain a lot of the other requirements of the gene sequence. And then we decided to first use a plasmid. So we cloned the genes into a plasmid, and then we induced the clone. And as you can see, these red dots are actually fluorescence, Nile blue fluorescent stained fluorescence microscopy images that show that we were able to make a PHA. And this is a more a high resolution image, a fluorescent microscopy image of the same. So we knew that we are able to use this strategy to make polyhydroxyalkanoate. Now the next step for us was how, and we actually thought we might be able to make polylactic acid, but eventually we ended up seeing that we had polyhydroxybutyrate and we didn't have polylactic acid, and we had to play around with the substrate in order to make polylactic acid. So we used a very rigorous extraction protocol from the chemists, and we eventually ended up after, I think, 72 hours with this evaporated condensate in this round bottom flask, which looks very plasticky when you see it, right? So this was the polyhydroxyalkanoate that we were forming. And then our first question was, okay, what are we forming here? It does look like plastic, but it might be anything else under the sun. So polyhydroxyalkanoates, either lactic acid, polyhydroxybutyrate, polyhydroxyvalerate, etc., they have very characteristic FTIR signals. So we went for FTIR, and we qualitatively determined based on the CO stretching, the rocking, and the carbonyl stretch at 1700s, and the CH2 stretch, the asymmetric stretch at around 2900. We actually concluded that the polyhydroxyalkanoate that we were making was indeed polyhydroxybutyrate. So we also looked up literature, and we saw that there was no way that we could quantitate polyhydroxybutyrate, or polyhydroxyalkanoate quantitation is rather a very tedious process. So, as most scientists do, we decided to go and reinvent the wheel. And since we saw that Nile red used to stain cells very well that contained PHP, we decided to develop a method, a quantitative method using spectrophlorometry to actually measure the PHB that was being formed in the cell in situ. And then we, once that was done, because, I have a PhD in chemical engineering, the next thing I wanted to do was, okay, let's check what is the optimal substrate, right? So we looked at various carbon sources, and we looked at glucose, we looked at xylose, we looked at glycerol, and we saw that glycerol was the substrate that produced maximum amount of the PHA or the PHP. And that's kind of obvious because glycerol probably leads to maximum amount of acetyl-CoA. But this was a journey, a discovery for me and some of my students. So then the next question was that, okay, we have it on a plasmid, we're able to express it, we make PHP. But, as we all know here as metabolic engineers, a plasmid has an antibiotic resistance marker, and that makes it not generally recognized as safe. So it's not GRAS. And we really thought we should integrate this into the genome. So we tried. So as a control, we always used to have the recombinant genes. So the genes from those two organisms, we had put them two on a plasmid. And we would always express them simultaneously in addition to these synthetically optimized genes that we are also introduced into a plasmid in E. coli. What was very interesting was that when we had the gene sequence optimized for E. coli, we were able to integrate it into the genome. But when we use the native sequence from the sourced organisms, we were successful. We must have tried screening multiple colonies of E. coli, almost 100 colonies of E. coli, but we did not find a single colony that had the recombinant genes incorporated into the genome. So that also sort of provides a reason to use gene optimization whenever it's possible in metabolic engineering problems. Okay, so now we get back. So remember a few slides ago, I left you hanging that, okay, we have now created a metabolic network reconstruction. We have converted that into a stoichiometric matrix. So why do we need a matrix formalism? Is because we can compute cell phenotype using the tools of linear algebra and linear programming and convex analysis. But how do you actually do that, right? What do you do with these network reconstructions? A network reconstruction is essentially this on the left-hand side. You have the networks very well delineated. You have the pathways which are balanced in there. And on the right side, you have a stoichiometric matrix. And for the case of PHA, where we added new pathways, or in any case where we add new pathways, we include those pathways into the stoichiometric matrix as well. Right? So there are many approaches you can use to actually convert these network reconstructions into models. The approach that my lab prefers is the constraints-based approach. And the basic premise here is that you essentially eliminate all the behaviors that cannot be exhibited by a network or by the system by systematically imposing governing physicochemical constraints. So let's take a toy example where there's an organism which has only 3 fluxes, right? Even my initial 2-dimensional annotation had just 3 open reading frames. So here we have a system that's defined by 3 fluxes. We call it an unbounded space. It represents everything. It essentially represents the whole universe in some sense. We successively apply constraints, these physicochemical constraints that I talked about, that include a lot of those, very intricate details we use in network reconstruction, right? Stoichiometry, thermodynamics. And then we also apply other constraints like enzyme capacity, genetic regulation, concentrations, and kinetics wherever applicable. And we end up with a more bounded space. And now this bounded space is called a solution space. And this actually represents what exactly that organism of interest can do. So the network of that particular organism of interest, what it can do. And so essentially, and I borrowed this quotation from Sherlock Holmes and the idea from one of my previous friends and colleagues from the Paulson lab. This whole approach is summarized in this quote, which says, how often have I said to you that when you have eliminated the impossible, whatever remains, however improbable, must be the true. So essentially, the whole concept of applying constraints is kind of elegant and simplistic because you just do more experiments and those experiments can be added as constraints and you keep narrowing down the solution space till you get to the phenotype of interest. Now, there are many constraint-based methods that have been developed over the last 2 decades. And, what I will allude to in this presentation is probably a little bit of phenotypic faceplane analysis. And of course, I think the second speaker today, or was it the third, he really gave a very good overview of flux bands analysis. And the fact that you have a system of linear equations, an underdetermined system, and in order to solve that set of equations, you need to call an optimization problem. We need to maximize something so that you get a feasible solution. And the optimization problem is based on the evolutionary objective of a cell to maximize growth. And that is why we look at the main objective function of maximizing growth. So if you look at a phenotypic phase plane, what exactly is it? It's actually a two-dimensional region, and it's spanned by two metabolic fluxes, and they are typically uptake rates. There are lines on this plane that demarcate the phase of constant shadow price. Now, when you call in the concept of optimization, of linear optimization, then you have both a primal solution and a dual solution. And when you look at the sensitivity analysis, you get two parameters there called shadow prices and reduced costs. Now, shadow prices effectively tell you what is the impact of a metabolite on that objective function. Like if you're maximizing growth, it will tell me what is the impact of, say, succinate or glucose on that particular objective function of growth. So by definition, the metabolic pathway utilization is different in each of these planes. So this plane of metabolic flux A versus B is divided by these three lines, and there are multiple regions over here. And so depending on the shadow price, you have different metabolic phenotypes. Like for example, in shadow price A, you might be making a particular product like acetate. In shadow price B, you will be making a metabolic product like succinate. In some, you might be completely oxidizing the product. So there are many characteristics of these phase planes, and some of the regions, the ones that I showed you earlier that are infeasible regions, the fluxes don't balance here, so you can't really apply the flux balance analysis philosophy. There are regions of single substrate limitations. There are regions of dual substrate limitations, and there are regions where there are futile cycles. So you're actually wasting a lot of energy here. There's ATP loss. Amongst all this, there's what is called the line of optimality, which corresponds to a maximal biomass yield, which is generally found by fixing carbon uptake and optimizing for biomass using FBA. And you will get 1 point in the line of optimality over there. And it essentially represents as one of the most important lines or concepts for metabolic engineering, because it tells you what is the maximum biomass yield that can be formed in addition to your product. So long ago, one of my colleagues looked at the optimality, the growth optimality principle, and studied the growth of E. coli on mallet. So what is represented here is a 2D projection of the phase plane with mallet uptake as the x-axis and oxygen uptake on the y-axis. And now the phase plane is divided into 4 regions. This phase 1 here represents metabolic futile cycles. Phase 2 is characterized by acetate overflow. The line of optimality, which is in red, separates phase 1 and phase 2 and represents the highest growth rate. The line of optimality on a phenotypic phase plane, as I told you earlier, corresponds to conditions of OUR and SUR that can maximize your biomass yield. And in those set of experiments, my colleague looked at growth of E. coli on glycerol. And although for mallet, my colleague found that the growth of E. coli was optimal, for glycerol, he found that the growth was suboptimal. So what he decided to do, or what my previous lab decided to do, was to subject E. coli to the selection pressure of this particular substrate. And on subjecting to about 600 to 1,000 generations of growth, they found that they could move from suboptimality to optimality. Now, why is this old result from maybe 15 years ago useful to me? The reason I find it useful is, as I told you earlier in one of my slides, Oops, maybe this is not the most efficient way, but glycerol, growth on glycerol for this genome integrated strain of E. coli made maximum amount of PHP. And so for me, glycerol became the selection pressure. And over the last, several months, my students were engaged in a process of serial passage during exponential growth, which is essentially the concept of adaptive laboratory evolutions that I think most of you are also familiar about in the field of metabolic engineering. And we used two selection pressures. One was glycerol, of course. We looked at the wild type as well because we wanted a control. We call the first strain SSE1 and we call the second strain SSE2. And why we have two different selection pressures here is that the plasmid that we used for a lot of the recombination had an arabinose PBAD promoter. So we are not actually sure because we have sent out our strains for genome sequencing, but we don't have the results yet. We are not sure whether the promoter has actually been incorporated into the genome. And we don't know whether arabinose will have any effect on the growth and product formation. So we decided to use both glycerol and arabinose either individually or together as selection pressures. And then the story just began for my latest. All this work until this point was done by a graduate student who's now graduated and he's moved on. He's a postdoc with UMN in University of Minnesota and NCBS. There's a joint Indo-US collaboration going on there. And I just found out today that he has had twins a couple of days ago. But this is where the story began for my newest PhD student, where she had some basal information from the previous student's work that this genome integrated strain indeed made some PHP and these strains had certain growth rates. So they have evolved, my student evolved these strains, subjected them to adaptive laboratory evolutions for 40 days. This corresponds to around 300 generations approximately. And as you can see here, the three strains show different growth profiles when you begin. And, they are changing across different evolutionary days. On day 18, they all seem to coincide. And then again, there seems to be some, deviation. And they are again on day 40, they are slightly apart. We don't know why. So this is now a new graduate student's project. And what she will be looking at is, are the glycerol uptakes different? Are the arabinose uptakes different between the wild type and these evolved strains? Are there any genome changes that affect these growth profiles and product formation? So this is all preliminary data that we had. Unfortunately, we had to stop experiments because of the lockdown in March. But my students are going to be back next week and I'm very excited about that. So maybe in the next on-site meeting, as Amit just mentioned after Shireesh's talk, I might be able to show you more recent results. And so we essentially, the bottom line is that we do see an increase in PHP. We do see a change in growth rates. What we want to understand and probe is the age-old question: does a cell optimize for its growth rate or its growth yield? When you are maximizing for your growth rate, when you optimize for your growth rate, then is there a change in PHP production? Is the yield? Do you get better yields of PHP when you have slower growing cells? So these are all the questions we are trying to explore in the coming months. So the summary and conclusions of this work are that synthetic and systems biology approaches are a common denominator now for metabolic engineering, allowing for scalable methods to increase the yields of any desired metabolite. And here I've given you an example of PHP. We can use a lot of modular strategies that are scalable to any other monomer in the case of these bioplastics. And so our next attempt would be to look at some of the higher chain fatty acids and their conversion. Adaptive laboratory evolutions have become a mainstay as well in metabolic engineering studies. And COBRA, or constraints-based reconstruction analysis, can really be used to understand the redundancies in metabolism. We have some other data where we've looked at the cofactor balance as well. So NADPH is limiting in these cases when you look at NADPH versus when you look at the competition between a substrate and the cofactor. So these are all things that we will be incorporating in the following, chapters of this study. And complete without acknowledgements. The most significant funding acknowledgements are to DST, DBT, CSIR, and the Gates Foundation. My collaborator in NCL, Venkat Panchagmula, who helps with all the metabolic analysis. My collaborators from Oxford Brookes, David Fell and Mark Pullman, who I bounce off ideas all the time, and Stefan Schuster from University of Vienna. And on the kanamycin project, I would like to thank Krishna Mahadevan from University of Toronto. This was my old network. All these students have graduated and have moved on. Mayur here was instrumental in developing all the genome integrated strains of PHA. And now we have evolved to a new network here, a new network state. And Shikha over here, Sakina and Tenyat were doing a lot of the laboratory evolutions. Sakina and Tenyat have moved on, so we have Shikha who will be taking this work forward. And yeah, so last but not the least, we have, that from rational strain design, if you really want to move on to a microbial cell factory, you need to scale the process, right? From rational strain design, you need to go to a shake flask, and then eventually you need to go to pilot plant, and then you can go to industry. You can make it a real microbial cell factory. So for this process, it generally takes a lot of time, just like drug discovery and drug development. This is also like a 10 to 15-year process. So in order to sort of accelerate it, we at NCL, at the MICE lab, we have just purchased and invested in this equipment. It's a microbioreactor system by M2P Labs, and it has a lot of online monitoring capabilities. Control fed batch processes are possible because of the microfluidic channel. So I invite any of you to NCL to work on this machine as collaborators, or if you just want to come down and play around with it, feel free once the coronavirus allows us to move around. And as the last slide, I'd just like to leave you with what our lab does apart from looking at metabolic engineering. We work on a lot of cancer projects as well and look at drug resistance, both antibiotic as well as chemotherapeutic. So with that, I would like to end my talk and would be happy to take any questions. Thank you. Thank you for your attention.
Thank you, Dr. Raghunathan, for a wonderful talk. It is really nice. It is a very motivating talk about the plastic production, PHA and PHB. So one person is asking, what is the current status of the bioplastic potential in India? So how it is India's point of view?
India's point of view, okay. That's an interesting question, a very useful question that generally funders ask me. All my funding agency asked me that, and I have to say that this project is currently not funded. So I haven't really paid a lot of attention to the current state of the art in terms of, what is the cost of production of the process, because also, as I mentioned in the beginning, I started off mainly from the point of view of basic science. How do you create a rational design? So I haven't really looked at it, I have to be very honest. But I know that in IIT Bombay, there's a huge effort going on. And I had put down that information somewhere, but I don't remember it top of my head. That I think from 1 gram of the substrate, I think they are able to go to up to 0.45 grams of bioplastic, if I remember right. But please don't quote me on that, because it's something I just don't remember top of my head. Maybe my student does, but there's no way someone on YouTube can comment, right? So maybe Shikha, if you know the answer, can you please type it in the YouTube box for viewers or for the person who asked me the question? Because we have looked into it, but I just don't remember it top of my head.
Prabhat Kumar Swain asked this question, so you can reply him.
Okay. Yes. So maybe we can take it offline, Prabhat. Thanks for the question.
Okay. The next question is about the acetyl coenzyme A, how the engineer pathway is diverting the acetyl coenzyme A. Is it?
So currently in our first generation strain, we actually do not have a engineered diversion. But in the future, we are planning to knock out the LDH so that we can get more pyruvate and pyruvate 2 acetyl-CoA. In the current generation strain that we have, it's mainly probably because of some mutations that have taken place that we are able to make more PHB than the original strain. Because if you see the genome integrated strain, when we started off, it produced very small amount in nanograms of PHB. So now we've moved to, I think, about 3 or 4 micrograms. So there has been a significant increase. But I think we need to look at what are the genetic changes that have taken place. And probably we will find some mutations in these genes that actually control flux to acetyl-CoA. But we'll probably know that in the next edition of this talk whenever I give that. So currently, we don't have an idea of we can only hypothesize based on our modeling approaches that we are getting more flux towards pyruvate, and that's probably making more acetyl-CoA. And the TCA cycle is probably diverting it through the acetate citrate lyase or something like that. Yeah. So
does that the acetyl coenzyme diversion reduce the growth rate?
Yes, it does, because we have seen the rates are decreasing, but the yields, we don't have much consequence on the yields because we let them grow for very long periods of time. But definitely there is an impact on the growth rate. So the growth rates are different. Yeah, they're slower.
So one person is asking that is Nitesh, that can we digest bioplastic?
Can we digest? Digest as in our body? I think there are some degrading enzymes. So there are some degrading enzymes that exist that can actually, I think they are depolymerases. So you'll depolymerize to the original monomer, which can be then probably recycled in regular metabolism.
I think he asked about whether the microbe can digest or not. Means most of the plastics are not reusable, or it remains as it is.
Okay. Yeah, so I think only if there's a specific depolymerase enzyme, I think people are I think the IIT Bombay effort does look at that particular enzyme as well. And they are moving towards creating a strain that can actually digest any plastic, not just, bioplastic, sure, but the regular plastic, if they can depolymerize it, then that should be helpful. So I think they are working on that. Yeah, we have not paid particular attention to that as yet, but that's something that's on our mind as well.
So Aparna is asking, like, is bioplastic as tensile as the normal plastic we are using?
I think bioplastics, especially the ones that we are looking at, the PHB and the PLA, they are very ductile and malleable, and they allow us to. You can make sutures out of them, and they will degrade in the body. You can make these bags, plastic bags. So I think their tensile strength is probably quite similar to, but we haven't really done those experiments in house. But we have a very strong rheology group in NCL, and they have told me if I get the first 20 grams of plastic, and they'll do all the rheology for me, because they really need higher amounts based on the way they do experiments to actually test a lot of these ductility and tensile strength, etc.
So you have shown the production of PHA as a glycerol as the substrate. So somebody is asking that this strain favors the conversion of glycerol to lactate, or when it is only produced when glycerol is used. Considering the glycerol inhibition in E. coli, is it a good idea to try a co-culture technique where one strain producing lactate and the other strain is utilizing the same for PLA production?
That's actually a very good question, and we definitely can try it. But as I know that we now do a lot of what do you call those metagenomic models as well, so where you can have multiple organisms in the same, you have a pan genome model and you can probe. So until now we've been only looking at pure cultures. So yeah, maybe it's a good idea to look at co-cultures in the future.
I have one question. The glycerol is a major waste in the industry, so can you use this glycerol waste from the industry for production of plastic?
Yeah, that is definitely one of our ideas. We want to move from using the pure substrate. So earlier I used to use a lot of glucose, and then we moved on to using, starch and some of the cellulosic, lignocellulosic biomass. So for glycerol, definitely we will be looking at some of the industrial waste, some of the biorefinery waste, because only then it will be a commercially viable process. So yeah, so we will be moving on to that. And now with the biolector on our hands, I think we should be able to high throughput a lot of these experiments.
For how to use the LCA study for bioplastic, and the LCA study or till date, anybody has done?
What is LCA? I'm sorry.
LCA is basically analysis.
Logical control analysis, is it? No.
It's a cost analysis, I think.
It's a cost analysis, okay. Yeah, I'm sorry, I won't be able to answer that question.
Okay.
I'm into spending. I spent like several crores buying the biolector. I need to now do some, calculations on the capex, etc. So yeah, so I think these are all very good points raised by the audience today, and I probably will keep that in mind when I come up with my talk in the second version.
Life cycle assessment analysis. So basically.
Okay. Yeah, so I think you have to do a capex analysis there, right? So, hello. Yeah, I think you're breaking up. I couldn't hear you. What did you say?
LCA stands for life cycle analysis, and which is generally done to actually.
Okay. Yes. Yeah, I think I'm not yet at that stage that I can do that. I think once I have a really good producer, I'll be motivated to do a life cycle analysis.
Yeah, this strain is only producing in milligrams. So once it produces in grams, grams, I think that
exactly
process optimization.
Yeah, then there'll be more motivation.
Yeah.
Exactly. Yeah. Hopefully in the second edition of the talk.
Yes, sure. Pradipto is asking for codon optimization process. How will we perform the gene will be expressed at significant level. So how you optimize the process, optimize the codon optimization, so optimization so that you have more gene protein level inside the
Uh,
cell. What's the limiting
so
factor?
So we essentially just used a existing algorithm where we keep changing the sequence of E. coli and then keep calculating how close the translated product is to the E. coli codon adaptive indices, because E. coli can use certain combinations of codons to only, right? It doesn't use certain codons. So we essentially kept an algorithm. It was an iterative algorithm where we kept changing the codons and calculated when it hit the maximum similarity to the E. coli genome. And that's when we actually stopped. So it was quite a long drawn out process.
Did you play with the promoter and terminator as well, or you just fix that one?
We played with the promoter as well. Yeah, so we identified that the PBAT would probably be a good choice. So we did some promoter, prospecting. Uh, but then we knew that eventually we wanted to integrate it into the genome. So it would not be as important as the actual gene. And what I thought was really interesting is that when we used the native sequences, we were never able to recombine it into the genome. They always got chewed off. I mean, we must have screened like crazy for several months. The graduate student was on his edge. But we could never find a single colony that actually incorporated those genes from that Acetobacter pastorianus and uh, Chromobacterium. So yeah, yeah.
So you have the state of the art facility right now, microfluidic setup, and you can generate new technology, new
Yes,
method.
I'm looking forward to it. I'm waiting. So yeah, so our installations got stalled because of COVID lockdown, and nobody's traveling, right? So we are
and
hoping that at least now, within a month's time or two, we should have the whole facility up and running.
it will be happy to learn from you regarding the
Sure,
microfluidic.
sure, sure. Absolutely. Would be happy to share.
And also,
And yeah, it's not a joke. If anybody wants to come down and use the facility, they are more than, it's after all government money.
yeah, and then we really interested to know your research in cancer cell, multi-omic systems biology, in how you're using in cancer cell and glioblastoma, and then the other antibiotic research you're doing, which is very interesting.
Sure, sure.
So
Yeah, that will be
our next workshop will be on systems biology, so you can speak on that.
sure.
So,
Absolutely. It would be a pleasure.
in this Spark proposal, we have to organize 2 workshops. So this is the first one.
I see, I see,
So
I
the
see.
next
Nice,
workshop,
nice.
hopefully, we'll have an in person or offline
I see, I see,
workshop
I
where you
see.
can actually come over in Kharagpur and present your work.
Yeah, that'll be nice. I think I visited Kharagpur 20 years ago, so it would be nice
Oh,
coming back.
yeah, great. We'll have a good interaction, and the student will also get a better interaction when in person happen.
Absolutely. Thanks for the invitation.
Okay, thank you, Dr. Anu, for a nice talk. We really enjoyed your talk, and I want to look forward to more interesting results from you in terms of microfluidics, then antibiotics, and then the cancer research you are doing. Thank you
Thank
so
you.
much. Thank you.
Thank you so much for hosting me.
Thank you for accepting the invitation and giving a nice talk. Bye. Have a nice
Bye.
evening. Have a nice evening.
You too.
So our next talk is from Alisa Walden from University of Washington in St. Louis. She is a senior student in Professor Tang lab, and she is almost submitting her thesis in few months, and she will be presenting her work on Aerolipilitica, the metabolic engineering work in Aerolipilitica. The title of her talk is basically biosynthesis of terpene compound using non-model yeast, Aerolipilitica. So Aerolipilitica, is actually very good in production of so many compound because uh, it is a very robust organism, and it's a non-model yeast where metabolic engineering is very difficult. So she will be talking about a very important subject today, how to do metabolic engineering in non-model yeast, which is very hard to do it because very less molecular biology is known. So it will be a very interesting talk from Alicia. So Alicia, you can carry on.
Okay, great. Thank you. Let me share my screen. And can you hear me okay?
Yes.
Okay, great. Okay. Okay, great. Well, hi, everybody. Thank you for having me. I am from Washington University in St. Louis. And as mentioned, I'm in Professor Tang's lab. I'm a third-year PhD student. And my work is centered around the synthesis of terpene compounds from Yersinia lipolytica, and kind of all that it entails. So just a little bit about me. I got my undergraduate degree in biomedical engineering from the University of Missouri. And now I'm getting my PhD in chemical engineering, again, from Washington University. Okay. So, yeah, basically just I'll give an intro to the topic and then go into 13C or carbon-13 metabolomics, which is the tool we use to get a better insight into the metabolism of Yersinia lipolytica. As mentioned, also it's non-model, and so not a lot is known about it. And then I'll go into what's coming up for us in the future. So terpenes are, if you're not familiar, are a large class of naturally occurring hydrocarbons. They're found in many plants, some insects, and they're used for a variety of different purposes, from fuel, biofuel, to fragrances and medicine. So it's really important to find a sustainable system to produce these compounds, so we're not cutting down trees and doing non-renewable methods of harvesting them. So my work centers around, or my project rather, will kind of consist of two parts. So I have been working on characterizing Yersinia lipolytica. We use strain W29. I saw a couple people presenting about Yersinia, and it is an up-and-coming species. A lot of people are working with it now. So yeah, I've been doing metabolic characterization using carbon-13 labeling, and then I will down the line work on process optimization to increase the titer of our desired products. Most of my work so far has been with a strain that's producing beta-carotene, but which is found in carrots. It gives it that orange color. But I will be starting shortly to work with two other strains that produce lutein and astaxanthin. Lutein is a drug for vision, and astaxanthin is a powerful antioxidant. So an overall goal of this project is to work towards the development of a sustainable system to produce these terpene compounds, as I mentioned. And one great thing about Yarwaya is that it's capable of growth on cheap feedstocks and industrial waste oils, making it an even bigger host of interest for economic reasons. So some other benefits of Yarwaya is that it has a high natural abundance of acetyl-CoA, which is the precursor to its native mevalonate pathway, which produces the precursors needed for terpene production. And as I mentioned, it's able to grow on cheap oil substrates. So our hypothesis with this work going into it was that upon feeding an oil-based feedstock to the cells, fatty acids are consumed and they produce acetyl-CoA through beta oxidation. So feeding a lipid would, in theory, increase the intracellular concentration of acetyl-CoA, which we hoped would increase flux towards the mevalonate pathway and thus increase terpene titer. And that was kind of our basis for wanting to work with oil-based feedstocks, as well as their inexpensive nature. Yarwaya lipolytica is also considered an oleaginous yeast, which means it has a high accumulation of lipids, just naturally. So this is a pathway map that I made for a review paper that I wrote, and it covers a couple different hosts, but Yarwaya would fall into this mevalonate pathway. Again, it's the mevalonate pathway is native to Yarwaya, which would reduce the metabolic burden of not needing to engineer this into a strain for production of these two terpene pathway precursors. So again, a fatty acid upon consumption would undergo beta oxidation and produce acetyl-CoA, and then hopefully continue on increasing the flux of these downstream pathways. One thing that is unknown, and I'll touch on this later too, is the compartmentalization of pathways in Yarwaya. So not a lot is known about how the transfer works from acetyl-CoA from the peroxisome to the mitochondria or to the cytosol. There's some genes that are known in Saccharomyces, and I think ACL has been shown to be there in Yarwaya, but that's one of our goals in this work too, is to just elucidate the metabolism and the compartmentalization of different pathways in the cell. So as I just mentioned, that's our biggest project aim, is to understand the complex metabolism of this species. Since it is non-model, not a lot of work has been done. And since it's a eukaryote, it has additional complexities like the internal organelles, the peroxisome and the mitochondria. So we want to know where the acetyl-CoA is generated and how it moves between the different compartments. And we're hoping that that will help us inform on engineering for the production of terpenes and to increase their production. Also, when growing on oil or lipid by itself, we're curious about how it produces the energy molecule NADPH. And it's also up in the air of whether or not beta oxidation occurs in the mitochondria as well as in the peroxisome in this species. And it could be different from strain to strain as well. Like I said, we're working with W29. And so these are kind of the big questions we're hoping to answer with this work. So as I said in the beginning, what we've been using is carbon-13 metabolomics to study the metabolism of this species. And a couple of people have talked about it, so I won't be too in depth here. It's pretty simple. So we just grow cultures on fully labeled or positionally labeled carbon substrates, and we do that in minimal medium, so there's no amino acids present, and so there's no other carbon source that would confound the results. Then we analyze those samples with a mass spec. With the GCMS, we analyze amino acids. We're able to derivatize those and analyze those on the GCMS. And then the LCMS we use for the analysis of free metabolites like G6P and citrate. So far on this, and then the mass spec puts out different peaks for carbon 12 and carbon 13, depending on how many carbon 13s are in that molecule. These mass ratios will be different. And from that, we're able to determine which substrate contributes mostly to which metabolites and with which pathways. So I've done 2 different kind of classes of this work, and one of those was co-carbon growth, so using 2 different carbon sources to analyze the metabolism of Yerwaya, and then also a pulse trace analysis. So for the co-carbon growth, we were looking at the analysis of amino acid labeling. And here what we did was we fed 2 carbon sources to the cultures. One carbon source was fully labeled and one was completely unlabeled. It's a diagram here. It shows a fully labeled glucose and then paired with unlabeled glycerol or paired with unlabeled oleic acid. And oleic acid is the most abundant fatty acid found in canola oil, which is an oil that we use. I'll get to that in a minute. So then we analyzed the resulting amino acids, and I took samples at the early log phase. So the results told us a good amount. We ultimately discovered that there's metabolic segregation, and we find that glucose and glycerol contribute more to glycolysis pathways, and then fatty acids contribute more to the TCA cycle. And that can be shown here. So in this graph, fully labeled glucose is used, and then the TCA cycle metabolites show low labeling when combined with a fatty acid or lipid. Also shown here, so we've kind of confirmed some results that other groups have discovered as well. The orange bar here is fully labeled glucose compared with or fed with unlabeled glycerol. And we see the labeling is really low, indicating that glycerol is a preferred carbon source to glucose when fed together. One thing that we did not find here is no catabolic repression and not a lot of gluconeogenesis, so or not a lot of trouble with it. So we also use fully labeled glycerol, which kind of shows compared with these oleic acid and canola oil that it's still a preferred carbon source too. And acetate we use as a short chain fatty acid and found that it mainly contributes to the TCA cycle compared to glycolysis with the pentose phosphate pathway. And we're determining this based on amino acid labeling from the precursor that is known to come from. So next I did a pulse trace study with inverse carbon 13 labeling with a lipid substrate, lipid-based substrates. I used canola oil, oleic acid, and glycerol. So what we did here was we grew cultures in fully labeled glucose and then pulsed them with unlabeled substrates, and then measured the dynamic changes over time. So the amino acid labeling is informative for steady state growth, but we don't get any information on dynamic changes. So this is the process of the pulse trace study, and I sampled from 0 hours to 24 hours. The results were informative again, and I put this back here for convenience. So we see that canola oil and oleic acid take at least 10 minutes to enter into the cell metabolism. This is likely due to the need for the production of surfactants and lipases, so that the cells are able to consume these non-aqueous substrates. We didn't use any detergent here. We just put it into the culture. Glycerol, on the other hand, is active nearly right away at 20 seconds in G1P and G6P and S7P, indicating that it's active in the glycolysis pathway and bendose phosphate pathway. So we see this again here, indicating that the M4 isotope log of citrate is active in oleic acid and canola oil after about an hour and a half. And then it's not really that active at all in glycerol. So this confirms that the fatty acid portion of canola oil, which is a lipid, contributes to the TCA cycle. And then on the other hand, the glycerol backbone of the fatty or of the lipid contributes to the to glycolysis in the pentose phosphate pathway, confirming our results that indicate metabolic segregation within the cell. We also performed biomass composition and cell imaging analysis. So here we found that in cultures with minimal medium and canola oil, and also in cultures with minimal medium and nitrogen limitation, we found a lower protein content as a percent of dry cell weight compared to the other cultures, which indicates that there's lipid accumulation occurring in the cell, where that's not happening in the other conditions. And although the protein content is different, there's no difference in the amino acid profile, which indicates that there's no changes in the protein expression between the different substrates. So interesting results we found here. We're observing in some conditions filamentous cell formation, which literature kind of agrees that it's strain dependent, and not all Yarwaya strains become filamentous during growth. But it's likely due to nutrient limitation or oxygen limitation during those growths. We observe here too that the filamentous cells can throw off the OD measurements as expected, and that in minimal medium, there's a lot of filamentous growth towards the end, indicated by a poor connection or poor relationship between dry cell weight and the OD measurement. So moving on to the beta carotene strain, the previous work was done with the wild type cells. Back to our hypothesis that oil-based feedstock will undergo beta oxidation and ultimately increase terpene titer. We've tested that with our lipid-based feedstocks, and also nitrogen limitation is known to increase lipogenesis in oleaginous yeast. so we attempted nitrogen limitation as well in an effort to increase the flux towards terpene titer. So it's known that beta-carotene will partition to the cell membrane and cause stress to the cell. And it also partitions to lipid bodies within the cell. We've seen this in literature and in our own work, little clusters of beta-carotene partitioning to lipid droplets within the cell. And here you can see the difference between beta-carotene cells and wild-type cells in minimal medium and in rich medium. And you can see the orange droplets from the beta-carotene accumulating in those lipid droplets intracellularly. I've also found during my work that the canola oil that's not consumed also will extract the beta-carotene from the cell, and then it'll accumulate in this lipid layer and make it easier or harder, depending on the process that would be used to extract the product at the end of a fermentation run. So ultimately, we found that beta oxidation of fatty acids contributes more towards biomass production rather than products generated from acetyl-CoA. And we also determined that carbon to nitrogen ratio is a really important factor for terpene synthesis and something that we need to look more into, because we see here that during canola oil growth in rich medium, the nitrogen supplements have a big effect on the overall production of beta-carotene. However, the beta-carotene per cell is still lower than conditions with glucose and minimal medium with canola oil as well. So coming up in the future, I will be working on carbon-13 metabolic flux analysis. And I'll kind of go over the overall challenges for this this project. As I mentioned, the morphology change is something that we're looking more into and seeing how that'll affect the metabolism of the cell, as well as genetic instability is common in fermentations, and that'll be something that comes up later in my work when I'm doing process optimization. And then all of these challenges with this project can be addressed in a few different ways. So metabolic flux analysis is the first step in what I'll be doing to address these questions that we have on the metabolism of Yarwaya. So we have a pathway map, and what we'll do is analyze the labeling profiles of as many metabolites as possible and constrain those with known factors of Yarwaya and end up with something like this, a flux map based on the substrate used. This is from a different species, but this is what we will be doing with Yarwaya, and we'll be including the peroxisome and the mitochondria compartments in that analysis, and hope to elucidate more information on the localization of different pathways in the cell. So to do this, I will be using positionally labeled substrates, whereas previously I was using fully labeled substrates, and oleic acid and palmitic acid are abundant fatty acids found in most plant and animal fats. So tripalmitin and triolein are the lipids of those fatty acids. I'm going to also be using a positionally labeled glycerol. And then these experiments will be analyzed on GCMS and LCMS for the amino acids and free metabolite labeling patterns. And then that information will be put into MFA modeling software to develop that flux map. So with this, like I said, we're hoping to determine pathway compartmentalization in the cell, and then from that, hopefully inform engineering of terpene producing strains to further increase the titer of those valuable terpene compounds. And then we also hope to gain an understanding of BRY's metabolism during growth on lipid-based substrates, which has not yet been elucidated. And this diagram does a pretty good job of showing the workflow of those experiments. It's not unlike the process I showed a few slides back. So of course, we're going to have some challenges with this work, particularly between distinguishing the intracellular locations of the metabolites between different compartments. It's really important that the constraints that we use are not randomly assigned, and they probably can't be based on Saccharomyces because ERY is a non-model species and isn't highly similar to Saccharomyces, and an incorrect constraint could really negatively affect the flux outcome. Also, it's going to be an issue combating the low signal of the positionally labeled lipids and fatty acids, as there's only 3 out of 56 carbons that are labeled, and the signal will be really low. So we're going to have to hope that it'll still be able to be distinguished and see what the patterns are for consumption of these substrates. Another thing that's challenging is the pre-existing pools of metabolites in the cell, and how that can confound the results that we'll see. And pool sizes with the labeled substrates are can be impact the results of the labeling results in the MFA flux analysis. So that's what my project is, and I'd be happy to answer any questions if I can about what we're working on for understanding the terpene production in this non-model species.
Hello, Ms. Warland. Thank you very much for your excellent talk on this non-model yeast. You said a lot about the metabolic engineering in Yerobia lipolytica. And we do have a few questions. So okay. Some of the people, they are asking, is there any selective reason to choose time from 0 hour to 24 hours in carbon labeling metabolism?
Yeah, so we need the time equals 0 to see what the breakdown of the labeling patterns were before we fed the non-labeled substrate, just to see the baseline of the fully labeled or close to fully labeled cells. And then 24 hours, that's typically when consumption has been enough to see the effects of the substrate on the metabolism. You can see kind of down here at 10 and 24 hours, they're pretty much the same labeling profiles with some exceptions. and so that's kind of why we choose those times. That's the window of the changing metabolism.
Thank you. So there is another question. How do you differentiate between engineering terpene producing strains than other strains in your study?
So I'm actually not the one doing the engineering. I'm just characterizing their metabolism once they are have been engineered, and kind of seeing if there's any other improvements that we can make in terms of metabolism flex, and wanting to increase flex towards the terpene pathway and decrease flex towards competing pathways. So in terms of differences, I guess, between the lutein strain, between the beta-carotene and astaxanthin strain, is just how long the pathway is, and how much extra metabolic burden is added due to that. So the lutein strain actually grows quite a bit slower than the beta-carotene strain because of this metabolic burden that it puts on the cell.
Thank you. So I don't think there are other questions. Thank you, Ms. Warland, again for the wonderful talk. And I think we'll have to wait for the final speaker for the day.
Okay. Well, thank you so much.
Thank you very much. So Professor Arul Mori will speak at 8 p.m. Indian Standard Time. So I think we'll have to wait for him. Thank you.
Amit.
Good morning.
Good morning. Good evening there.
So a lot of people attended in the morning. In the evening talks, we are not that very. Not many people are watching now.
Not many are coming. Okay.
Now it is online. So Professor Arul is back. We will start the session now. A lot of people attended in the morning. In the evening talks, we are not that very. Not many people are watching. So I'll So let's begin the session. Let's come. Have a 15 minutes break, and now we should be back to our workshop, and we'll start the last talk. The last talk will be given by Professor Arul Varman from Arizona State University. So he has been our Spark collaborator and has been working on metabolic engineering systems and synthetic biology for production of biofuel and pharmaceutical and value-added chemical. So mainly he is working on cyanobacteria, which is very important organism in terms of bioproduction using sunlight, which is very useful in India. So he holds a master degree from chemical engineering from IIT Bombay, and also a recipient of Department of Atomic Energy Fellowship. And his research and his PhD was from Washington University under Professor Tang, where he has done PhD in metabolic engineering for cyanobacteria, for value-added chemicals, and a lot of work he has done there. And then after doing PhD, he moved to Berkeley, where he has done his postdoctoral work in CSIR-National Lab, and as well as in Joint Bioenergy Institute under Dr. Seema Singh. So after completing postdoc, he is an assistant professor in Arizona State University, working on different important organisms like cyanobacteria and many other organisms he has. In today's presentation, I welcome Professor Arul Varman and request to give this talk today.
Good evening, everyone to India. Good evening to everyone in India. And thank you, Dr. Ghosh, for the nice introduction. And thank you as well for inviting me to this excellent workshop. So this is good morning for people who are in the US. Hopefully some of my students are awake and watching. All right. So as Dr. Ghosh introduced, I work on systems and synthetic biology. And today I will mainly focus upon utilizing synthetic biology for the valorization of lignocellulosic biomass. And even yesterday during one of the talks, one student asked a question to the speaker, what do we do with the rice straw? Okay, instead of burning it, how do we use it, right? Or is there a better use for it? And as the speaker said, you can produce fuels and chemicals out of it. So this talk will take you, or this talk will elaborate on that direction. All right. So the outline of my talk, I will give a brief introduction on the motivation for this subject, and we will have an introduction to what metabolic engineering is for some of the newbies. I know that from the morning to afternoon session, you would have been exposed to different metabolic engineering and synthetic biology talks, but we always try to prepare it in a manner if there are newcomers for our talk, even they should be on board with it. Okay, so we will give a brief introduction to what is metabolic engineering here, or what metabolic engineers do routinely. And followed by that, I said the talk will focus mainly upon valorizing plant biomass. Apart from plant biomass, the other major carbon feedstock available around us is carbon dioxide. And I will give a brief intro to utilizing carbon dioxide for producing value-added chemicals. Followed by that, utilizing plant biomass as a carbon substrate for fuels and chemicals. And there will be a separate section on lignin valorization. It's still part of the plant biomass, but this is a developing field, so there will be a separate section on that. And ultimately, how do I mix all these up? How do I valorize both cellulosic, hemicellulosic components of plant biomass, as well as the components that I obtain from lignin all together in a single part? So we'll look at the technology that we are working in that direction. All right, so this is a slide I think plenty of you must have seen from today, and I will keep it short. So we know that we are getting fuel out of crude oil, but crude oil doesn't serve as a source for just a fuel, but source says that about 6,000 different products are being produced from crude oil. And if not sooner, but sometime in the near future, we will run out of crude oil. And of course, it has some environmental impacts as well. So we have to develop renewable and sustainable alternatives for the production of building block chemicals for all these different products. Okay? And that's where we metabolic engineers and the synthetic biologists come in. So we want to synthesize the building block chemicals as well as fuels by utilizing microbial cell factories and convert renewable carbon feedstocks that are available around us in plenty. And there are two major carbon feedstock that are available around us in plenty. First one is carbon dioxide, like I have mentioned in the introduction slide. So by growing these photoautotrophic organisms, green algae or cyanobacteria that can fix carbon dioxide in the presence of sunlight, we can produce fuels and chemicals by engineering their genome or by manipulating their metabolic pathway to increase flux towards the production of fuels and chemicals. So this is one major carbon feedstock that is available around us. The other major carbon feedstock that is available around us is plant biomass. Okay? So it has three major polymers in it. And by breaking down cellulose and hemicellulose, we can obtain sugars. And these sugars can be fed into microbes. And if the microbes are engineered, we could produce fuels and chemicals at a higher flux. And there are two, let me get my laser pointer. So there are two routes to valorize plant biomass. One is hydrolyzing the cellulose and hemicellulose components in a separate part, obtain the sugars, and those sugars are converted into fuels and chemicals in a different part. And there is another process called consolidated bioprocessing, where both hydrolysis and fermentation are done in a single part. And we are exploring both these routes in our laboratory. Okay, or in fact, when I take a look at this slide, so we are exploring all three routes, converting both carbon dioxide into fuels and chemicals, or converting plant biomass via these two different approaches into fuels and chemicals. And when we look at lignocellulose or plant biomass, there is one major component of lignocellulosic biomass called as lignin. It makes up, in some plant biomass, it makes up to about 30% of it. It's recalcitrant in nature. And so far, people have been looking at it as a barrier to obtain cellulose and hemicellulose. So they were using destructive technologies, break down this lignin, and usually it is burned for its heat energy. However, when we run out of crude oil, we'll be running out of sources for the production of different aromatics. And lignin has aromatics as its building block. And we see that by utilizing lignin as a feedstock, we will be able to produce several aromatics that will run out of soon. So we are focusing on lignin valorization as well. So what is metabolic engineering for some of the newcomers here? So if I ask what is a cell to most students, it is the tiniest living organism. But to a metabolic engineer, it's a microreactor in which thousands of reactions that are shown over in this slide take place. And when a metabolic engineer decides upon a product he or she wants to synthesize, what we do is we focus on the reactions that are the pathways that result into the precursor molecule for producing that product. So we zoom in there. And often what we do is we overexpress foreign genes or native genes to increase carbon flux towards the fields and chemicals of our interest. And I think this was one of the question to Professor Tang as well yesterday. So once you decide on the product, what all steps do you follow to increase the flux for your fields or chemicals? Okay, so these are the routine steps. So we overexpress foreign or native genes to increase the flux in that corresponding pathway. And not only that, we also knock out byproduct synthesis. A very common one is ethanol. And if we knock out byproduct synthesis, that carbon will be diverted to fuels and chemicals of our interest. So these are the routine strategies that we follow in metabolic engineering to increase fuels and chemicals. So this would all look like by using a GPS system, and we would want to travel from point A to point B, and the point A here being a substrate in my metabolic pathway, and the point B being fuels and chemicals. Okay. So this is a stationary system. Okay, so your Earth is not continuously shifting. But to metabolic engineers who have done these changes, they would have seen that this is a familiar moving staircase in the Harry Potter movie, right? So you want to land at one place, but as the staircase moves, you will land somewhere else. And that happens very often in metabolic engineering. Okay, so you expect something, and we land somewhere else. It can be for various reasons. It can be due to cellular stress or metabolic imbalance and so on. And herein we take advantage of systems biology techniques. So from genomics to metabolomics, and fluxomics is the one we have focused more about. And in the previous talks, especially from Professor Tang to my student and Dr. Ghosh, they have all spoken about fluxomics. So by utilizing all these systems biology techniques, we would understand what happens in the cell at the systems level after engineering. And we try to use that design, build, test cycle, engineer the organism further, and optimize it further and further for improving flux towards our product. Okay, so this is metabolic engineering in brief. So the first research section, how do I use carbon dioxide as a carbon substrate for producing fuels and chemicals? So for this work, we use cyanobacteria as a host. So shown over here is Synechocystis. It's a blue-green algae. So important thing about its metabolism is, along with it being photoautotrophic, that is fixing carbon dioxide in the presence of sunlight, it can do mexotrophic metabolism as well. So it can utilize organic carbon substrates along with carbon dioxide as its feedstock. And important for metabolic engineers, it has a fully sequenced genome, and it is naturally competent to uptake foreign DNA. So it's very easy to engineer than most cyanobacteria available around us. And it is faster growing than green algae. So this was actually my PhD work. But once I graduated, there were several other cyanobacteria that were discovered that were way fast growing than Synechocystis. Okay, so if I were to travel in time at the time, I would have picked one of those fast-growing organisms. In fact, okay. So they are much better producers of fuels and chemicals now. Okay. However, this still gives a general idea of how you could utilize carbon dioxide and convert it into fuels and chemicals. So the objective of this work was to produce D-lactate. So D-lactate is a precursor for biodegradable plastics by utilizing cyanobacteria. And same during my PhD work as another example. So we did engineer cyanobacteria for the production of isobutanol, which can serve as like a drop-in fuel molecule, right? So, but this work will focus on D-lactate because there is an interesting 13C fingerprinting study from this work. Okay, so as my student presented his talk, so he introduced about 13C fingerprinting. And I thought this will be a good example where you can see the application of 13C fingerprinting in understanding the metabolism of engineered strains. All right. So this is the metabolic pathway in brief that we are focusing on. As you can see here, for the production of D-lactate, the precursor molecule is pyruvate. And by introducing one gene that codes for D-lactate dehydrogenase, I can convert pyruvate to D-lactate, right? And you can see that this reaction, so for people who are not familiar with bio, so you can see this as a two substrate reaction along with pyruvate. It also involves another, what we call as a cofactor, all right? So in cyanobacteria, the abundant cofactor is NADPH, and for this reaction, we need NADH. And so we engineered this organism further by introducing a transhydrogenase that can convert NADPH to NADH, right? So basically what we do is these are these two genes that I spoke about that codes for D-lactate dehydrogenase and transhydrogenase. They are under the control of a promoter. So this is another small piece of DNA. So you can imagine as like a switch in your home, electrical switch in your home to turn on and off lights. So this is like a switch inside the genome to turn on and off the expression of genes, all right? So this is my design, and what we do is we try to integrate them into the genome of cyanobacteria here. And along with introducing these foreign genes that I want to overexpress to produce the chemical of my interest, I also introduce an antibiotic resistance marker. Okay, so this makes the engineered strain resistant to kanamycin. Okay, so once I transform cyanobacteria, then what I do is I plate the mixture of cyanobacteria to a plate containing antibiotics. So on the wild type that will be still present, they will die because they don't have this marker in there. But the engineered strains, along with taking the foreign genes of my interest, they would have taken up the kanamycin resistance marker. Henceforth, they become resistant to antibiotic. Okay, so I can select for my engineered strain by using this antibiotic resistance. And cyanobacteria, interestingly, has multiple genomes in it. Right? And so we have to integrate these foreign genes into all of its genome. And for this, what we do is we do a process called segregation. So we continuously plate these strains in the presence of antibiotic. And from Darwin's theory, survival of fittest, and here the fittest one would be the ones that have all of the antibiotic resistant marker integrated into all of its genome, right? So more antibiotic marker present in a strain, so it will survive better, all right? So using Darwin's theory of evolution, we continuously plate them into antibiotic plates, and we select for colonies that have the genes integrated into all of its genome. And those are called completely segregated strains. So once we obtain them, so we conduct our experiments. And as you can see here, this shows the D-lactate production with time. And as you can see here, the strain that is expressing the D-lactate dehydrogenase along with transhydrogenase starts producing D-lactate early on, and it accumulated about a gram per liter of D-lactate at the end of our experiment. And this experiment also proves the impact of increasing cofactor availability towards product synthesis. So to answer one of the students' question, there are several approaches depending upon the pathways that you are working on. So this is one. So Along with overexpressing the relative genes, you also should focus upon increasing cofactor availability. All right. And like I said before, this organism, the important feature of this organism is, along with taking in inorganic carbon substrate, carbon dioxide, it can consume organic carbon substrates. So when we try to grow this organism in the presence of glucose and acetate, so the wild type cyanobacteria grows about 5 times faster in the presence of glucose. So we thought that we will see this in the production as well. However, as I said in the Harry Potter slide, the opposite happened, right? So in glucose, the organism was supposed to grow faster, but the engineered strain did not perform well. In fact, it's not shown over here. After a few days, we can see that the strain was getting stressed, and it was what we call the phenomena called bleaching out. Okay, so the cells were starting to die, and there was no increase in the levels of D-lactate titer at the end of it. And interestingly, when we fed the organism with acetic acid, it doubled up the titer. Okay? So we wouldn't see that big a growth change, growth impact in the presence of acetate, but this resulted in increasing D-lactate titers by twofold. And so here we demonstrate by utilizing multiple substrates, we could increase D-lactate titers. And again, I simply say that utilizing multiple substrates, but you have to choose it wisely again, depending upon the metabolic pathway that you are engineering. Okay. So we wanted to understand the role acetate plays in increasing D-lactate synthesis. And here we did the 13C labeling experiments where we fed the organism with unlabeled carbon dioxide and labeled organic substrates. So in this case, we fed the organism with labeled glucose and labeled acetate. For people who are not familiar with labeling, so instead of the natural 12 carbon that is present, so certain atoms in the carbon substrate of our interest would be labeled with 13C, the heavier isotope, all right? And by using mass spec, we can track where this carbon transition or where this carbon flows through in the metabolic pathway of this organism. So like I said, we analyze them via mass spectrometry and get the abundance of the products of our interest and look at the mass spectra here, right? So if I consider lactate, a 3-carbon molecule, there are different isotopomers that can be present. So one of the D-lactate, a certain fraction of D-lactate could be unlabeled, and that mass fraction, we label it as M0, and some amount of D-lactate will have only one of its carbon labeled, and that we call as M1, and some can have 2 carbon labeled M2, and some can have all 3 of its carbon labeled, and that is 3, okay? So this in short, we call as a mass distribution vector. How is the mass for that compound of interest being distributed in the metabolite pool of the organism, right? So we look at the mass distribution of the product or precursors of our interest, and from which we make certain important analysis. Okay? For example, in this case, if I say that if I look at my mass distribution vector and see that there is a high 13C enrichment, and the way this could have happened is if the organism or through this pathway, more carbon from glucose and acetate have flown, right? So the labeling carbon from glucose and acetate have flown into the product. So this would confirm that the pathway the organism has undertaken for the product synthesis has borrowed carbon from the organic carbon substrates. And if it is a low 13C enrichment, it means that the organism is taking up more of carbon dioxide. In this case, that is the unlabeled substrate, right? So we did this analysis for both glucose and acetate. And I said here, it was labeled glucose and unlabeled carbon dioxide. Shown over here is the distribution of masses for lactic acid and several other amino acids. And you can see that the mass fraction for several amino acids increases, right? So this means that the carbon from glucose has flown through the metabolism, but for some reason that did not translate into the lactate titer. Okay? So we didn't do further investigation, but this is most likely to what we call as metabolic imbalance in the organism. Okay, so that didn't help the organism, and that didn't do well. But on the other hand, I said that lactate titer was increased by twofold in the presence of acetic acid, right? So when we fed the organism with labeled acetate, since in the presence of acetic acid, the titer of lactate increased by twofold, I would expect that the carbon from acetate flowed into lactic acid synthesis, but that did not happen. Okay, so this is a tiny bit of labeled carbon present in lactate, which can be neglected actually, because 1% of carbon that is present naturally is labeled. So this can be neglected, of course. And so most of predominantly lactate was still synthesized from carbon dioxide. So carbon from acetic acid was not being contributed for lactic acid synthesis. And when we look at amino acid synthesis as well, only 2 of the amino acids had carbon flowing from acetic acid. So this was very interesting. So acetate resulted in delactate titer improvement, but did not contribute carbon. So when you look at the metabolic pathway, so this is how the pathway looks. So acetate flows into acetyl-CoA and flows into TCA cycle, and several amino acids are synthesized from the TCA cycle, right? So in a way, if the carbon from acetate has to flow to delactate, it has to flow in an unfavorable direction. Okay, acetyl-CoA needs to be converted into pyruvate. Okay, it's not favorable. But so what could have happened, right? So we think that as we fed the organism with acetate, it improved acetyl-CoA titers. It improved the pool of acetyl-CoA present in the organism. So for this reaction of pyruvate to acetyl-CoA is the product, right? So as we increase the product titer, the forward reaction rate could have decreased, resulting in improvement in pyruvate titers. So as we increase pyruvate titers, okay, so by feeding the organism with acetate, indirectly with that, it resulted in improvement in the titer of pyruvate, which is a precursor for delactate. So for this reaction, as you improve the titer of your substrate, then the forward reaction is going to be much faster. And that's how we increased delactate titers indirectly by co-feeding the organism. So that's briefly upon how carbon dioxide was used for the production of biochemical. Next, we will move on to utilizing plant biomass as a carbon substrate, as a carbon feedstock for the production of several different chemicals. So there is plenty of sources around us for plant biomass. Agricultural residues, agricultural waste can be one major source for plant biomass. And when we look at plant biomass, there are 3 major polymers. And out of this, when we look at cellulose and hemicellulose, when we break them down, we can get 6 carbon and 5 carbon sugars that can be fed into microbial organism or engineered microbial cell factories to produce fuels and chemicals. Like I said here, lignin is another major polymer, but it has about 10 to 12 different linkages in it, making it very complex to be broken down enzymatically, and therefore usually chemical treatments are done. But we are also looking at lignin as a valuable source, and we are developing biocatalytic technologies for valorizing lignin as well. Okay, so we will focus on different components of plant biomass in this lecture. First, we will look at the hemicellulosic component. Right? And so I said that hemicellulose is broken down into its sugar in a different part by utilizing enzymes. Okay? So this enzyme purification step makes this whole biorefinery concept a bit more economically unfavorable. Okay? So we wanted to develop a strain that can secrete these enzymes and break down these cellulosic and hemicellulosic polymers in situ and release the sugars in the presence of the organism itself. So basically, we want to engineer this organism, secrete enzymes that can break down these polymers and release the sugars. And this organism in turn will feed on these sugars, and we could valorize the polymeric part of a plant biomass in one part. Okay, so this can be broken down into three objectives. First, we want to engineer the organism to secrete xylenases. So I say xylenases because, as a case study, I want to show in situ depolymerization of hemicellulose. Okay, by utilizing xylenases, I'll be breaking down xylene, which is a type of hemicellulose. Okay, so we'll be demonstrating depolymerization of xylene to xylose, and we would show one-pot demonstration of converting hemicellulose to valuable chemical, right? Okay, so this is the selection strategy in brief. So we cultured our engineered strains, and what we will do is we collect the supernatant. So this would be our first study to understand if the engineered organism is in fact secreting the enzymes of our interest. Okay, so once I engineer the organism, I could collect the supernatant of that organism. So the expectation is if the engineering worked well, so the supernatant will have the enzymes that will break down the cellulosic and hemicellulosic components. And in this particular case, the supernatant should be enriched with xylenase. And I do an in vitro hydrolysis reaction where the enzymes that are present in supernatant would break down xylene into xylose. And we take advantage of what is called as a DNS assay. So what it does is if monomeric sugars are present in the supernatant, so there will be a color change, as you can show here. Okay, so if the color changes, if the color becomes more darkish in color, then it means that that particular well is enriched with the sugar molecules, and we can pick the strains that are responsible for that well. So this was our screening strategy for isolating the best performing hydrolyzed strain. So my student, my PhD student Apur Matre was doing this work. So you must have seen in today's morning lecture. So he went ahead and constructed several different xylenase secreting strains, and we followed the screening strategy that I spoke about in the previous slide. And we screened the different strains under the different concentration of substrate xylene. And as you can see here, so consistently one strain performed better at different xylene concentrations. So we zeroed our efforts to this one strain, and we conducted further studies. So right now we tried breaking down xylene in situ. Okay, so in the presence of bacteria, we didn't separate the supernatant out. So the xylene was added directly to the culture, and we expect that the engineered bacteria will chew up the polymer and release the sugars in situ. All right. So shown here is xylene kinetics and the xylose kinetics. So over time, the best performing culture condition, we expect the xylene should go down faster. So we utilized a different inducer concentrations. So this is the promoter I spoke about. Okay, so I can turn on this promoter by adding these chemicals at different concentrations. As you can see here, at 0 millimolar concentration of this chemical, the xylene breakdown rate is not very fast, but as I start increasing the xylene concentration, it starts increasing, but after a certain concentration, it goes in the opposite direction. Okay, so we found out that 0.2 millimolar was the best. It accumulated about 6 to 7 gram per liter of xylose. Okay. And this is one of the best xylose titers reported via in situ depolymerization of xylene. All right. And next, we wanted to valorize this xylene. So we developed this strategy. So we wanted to put a food producer. So in this case, the xylose producer that we have been talking about, along with a product producer that will consume this xylose, and in this case, produce a succinate. Okay. So my collaborator, Dr. Wang, had an engineered organism that can feed on xylose alone and convert into a succinate. So we did the co-culture basically. So one of the organism will break down the xylene and the other organism will feed on the broken down xylene and produce this product. So in this work, we were able to demonstrate that we can engineer an organism and produce about 7 gram per liter of xylose, and that xylose in turn can be converted into another molecule called succinate at about 3 gram per liter, right? So in the previous work, we saw the efforts that are in the direction of valorizing the hemicellulosic component of plant biomass, okay? So similar efforts are underway to break down cellulose into its monomeric component glucose. And here we will look at one example of how this glucose can be converted into an useful chemical, in this case, ethyl lactate. Okay, so ethyl lactate has several uses in industries. It can be used as an biodegradable industrial solvent, plus we have several other uses. So it has a demand of about 5 million metric tons annually, right? So this is a simple reaction by having an carboxylic acid and an alcohol, so it can be esterified to form the ester. Okay, so these are the two precursor molecules we need for producing ester, okay, that you want to remember, okay? And so the goal of this work is to engineer an organism to produce ester in the presence of glucose, okay? So the challenge here is to isolate enzymes that can esterify this carboxylic acid and alcohol into esters of our interest. So my postdoc Aditya, he went and explored several different yeast species and mined for genes that will code for esterases. Okay, so he compared different esterases isolated from different sources, and he zeroed down to esterases of his interest. And this is the metabolic pathway that we are focusing on. Okay, so we'll have, for example, ethanol as one of the precursors for producing ethyl acetate or lactate. For ethyl acetate, you will have acetic acid coming in. And for ethyl lactate, you will have lactate and lactyl CoA coming on the other side, right? So with this, he constructed the strain that's shown over here to overproduce ethanol, lactic acid, and the enzymes to esterify the ethanol and lactate. Okay. So initial experiments, he isolated the strains that can accumulate ester titer between 3 to 4 milligram per liter. Right? So we did a further study. So we did high cell density cultivation. So more of microbial cell factories are in the culture, so they can churn out this ethyl lactate molecule much faster. And along with that, he did optimization of the inducers that we spoke about and culture temperature optimization. With all of this, we were able to improve ethyl lactate titer by about 2-fold. Okay, so this work shows how you can valorize the cellulosic component of the plant biomass into an useful chemical. Okay. So far we have seen how to valorize the cellulose and the hemicellulosic component. And I said that one of the major challenges when you look into a plant biomass is lignin, as it is recalcitrant in nature. But researchers world over are recognizing that this can be an important feedstock for producing aromatics. Okay, and in this work, we will look at an organism called Sphingobia SYK6, and it is known to have a well understood ligninolytic pathway. So we can engineer this organism to valorize lignin-derived aromatics. And this organism was isolated from wastewater plant associated with the paper and pulp industry. And as you can imagine, something that can survive in there must have fed on the residues and most likely the aromatics that came out of lignin. And we know that this organism can grow on lignin-derived monomers and dimers as its only carbon source. And important for metabolic engineering, it has its genome sequenced, and it is also amenable for genetic engineering. Okay, in here, we wanted to utilize 13C fingerprinting and throw light into the metabolic pathway of this organism. So we could engineer this organism by understanding its metabolism further. And we also want to do flux analysis to understand the rate of the central metabolic reactions. And that will also assist in engineering this organism rationally. And the transcriptomics is another systems biology technique, which can be used to confer our findings from 13C flux analysis, right? So 13C fingerprinting background in brief. My student must have spoken about this in the morning, but briefly for people who are appearing just for this talk, just in case. So 13C fingerprinting in brief. So if as glucose enters the metabolism, it can channel down through two pathways. It can go via the PP pathway or the glycolysis, all right? So to understand if most of the carbon flows via PP pathway or glycolysis, what I can do is label glucose in its first carbon. Okay? And if the organism has enhanced flux towards PP pathway, and as the molecule flows through the PP pathway, the first carbon gets lost in the PP pathway. And therefore, the metabolites that are result from the PP pathway will have unlabeling present in them. Okay, the labeled carbon would be lost. However, if glycolysis was highly active, then the labeled carbon would be conserved, and the metabolite pool will be enriched with the labeled carbons. Right. So this will say the relative rates of PP pathway versus glycolysis. Likewise, we can utilize several different labeled molecules and interrogate different pathways that are present in the organism, right? And so to do this study, we will look at a labeling pattern of amino acids. The reason we look at amino acids and not these intermediates of a central metabolite is because a protein make about 50% of your biomass, and these proteins are in turn made up of amino acids, right? So the pool of these amino acids is much larger than these intracellular metabolites. So that's easier for us to analyze these amino acids. And by looking at the labeling of the amino acids, we can say how these intracellular metabolites would be labeled. Okay? So we basically look at labeling of amino acids to make judgment of which pathways are active and not. Okay? So this is a simple 13C fingerprinting work first. So there was this hypothesis, or there was this rumor around that this organism is not good for feeding glucose. And in fact, one of the key reactions in glycolysis or glucose uptake was missing in this organism. But we always know that organisms have other backup pathways with which they can assimilate organic carbon substrates, right? So we wanted to interrogate and confirm if this organism indeed has glucose assimilation capabilities or not. Okay? So we fed the organism with labeled glucose in the presence of rich nutrients, and we looked at the labeling pattern. Okay? So M0, like I mentioned before, it indicates the mass fraction that is unlabeled. Okay? M1, M2, M3, and so forth indicates the mass fraction that is labeled. And as you can see here, the M naughts are more than 90% for some of the amino acids shown here. So owing to space limitation, I am not showing rest of the amino acids, but you can see here the unlabeling hasn't decreased compared to the control. And therefore we can confirm that the labeling from glucose did not penetrate the metabolism. So therefore this organism does not have the ability to uptake glucose. So this gives us, one other challenge. So if we want to valorize lignin-derived aromatics, we can also focus upon engineering this organism to uptake glucose as an organic carbon substrate. And parallelly, we can valorize the aromatics. Okay, so that's one possibility. And I mentioned that this organism can survive on lignin-derived aromatics, and that vanillin is one among the major aromatics that are derived. And we wanted to confirm if this organism can indeed take or do 13C fingerprinting and study to understand the flow of carbon from vanillin to the metabolic pathways. And you can see here the labeling or the mass distribution vector for different amino acids in the presence of labeled vanillin. You can see here the M0 values. In the previous ones were in 90s, and now it has decreased. All these higher EMs have increased. Okay, so indicating that the labeled carbon from vanillin has penetrated into the metabolism. So therefore, this organism definitely has the capability for producing or making its biomass components based on lignin-derived aromatics. All right. So another example of a 13C fingerprinting. So we did different labeling studies in this organism to understand the different pathways. And now we want to understand one pathway called as cataplerotic pathways. So this connects the TCA cycle to gluconeogenesis, and we want to understand if a cataplerotic pathway is present in this organism or not. So to understand that, we fed the organism with labeled acetate, right? So there are two routes by which carbon from acetyl-CoA can flow into pyruvate. One, by the direct conversion of acetyl-CoA to pyruvate. So if that happens, unlabeled carbon dioxide needs to be fixed. And therefore, when you look at mass distribution vector for pyruvate, M2 should be high, okay? So when we look at mass distribution or vector of alanine derived from pyruvate, M2 was not high, but it was M3 that was high, okay? So how did we end up having a molecule that is labeled in all three of its carbon, but we fed the organism with only organic carbon substrate that has two carbon labeled. So how can that happen? That can happen only when this carbon from the labeled molecule goes through a cycle and the labeled carbon gets accumulated and the cataplerotic pathways active and the pyruvate being synthesized. Okay, so basically the two labeled acetate flows in and it accumulates, the labeling accumulates in the TCA cycle intermediates, and as cataplerotic pathways are active in this organism, three labeled carbon pyruvate is being synthesized in this organism. Okay? And this confirms the presence of a cataplerotic pathway in the organism. And next, we wanted to understand the flux of central metabolic pathways in this organism. And the challenge here is lignin has several different linkages, and it can correspond to different aromatics as well. Okay, all of them through different catabolic pathways, they flow into vanillin or vanillic acid. Okay, so to do 13C flux analysis, we have to know in which position the substrate has been labeled. Okay? So if we select lignin as like the source, then we won't know from where the labeling has been derived in the organism. So we need to pick up one carbon substrate. Okay, so we picked up vanillin with the hypothesis that the different degradation rates of or depolymerization of lignin will just change the vanillin pool in the organism. And so we just fed the organism with vanillin to interrogate the flux of the pathway inside this organism. So like I have mentioned before, so we grow this organism and collect the samples in the exponential phase. That's where we assume a pseudo steady state, and we do mass distribution vector, or we obtain mass distribution vector by using analysis that was described by Arul in detail. And the mass distribution vector are utilized as constraints in a model. So basically, mass distribution vector acts as a storage repository of the relative rates of fluxes within the organism. Okay, so these can be used as constraints to obtain the relative fluxes in the organism. And we typically use a software called WooFlux developed in the Tang lab and obtain fluxes of the organism. All right. And like I said before, so we fed the organism with labeled vanillin, and we obtained the metabolic flux map of this organism. As you can see in this pathway, okay, so the thicker the lines indicate the flux through that pathway is very high. So as vanillin feeds into the metabolism, so its flux is pretty high. And you can see that the vanillin pathway contributes not only to the carbon molecule, the carbon substrate for the organism or the carbon building block for the organism, but it is also important in the synthesis of cofactors NADH and NADPH. Okay, so when we looked at D-lactic acid synthesis, we saw the role of cofactors. Okay, so cofactors are important for its biomass components as well. So this vanillin catabolic pathway is not only important to provide carbon for the organism, but it is also an important cofactor pathway as well. Okay, so this confirms the importance of vanillin catabolic pathway in the organism. And along with that, by utilizing 13C fingerprinting studies, we identified or we explored different pathways in this organism. And interestingly, we also found that this organism has a very high carbon loss, okay? As the carbon from vanillin flows to pyruvate and oxaloacetate, and oxaloacetate flows through the TCA cycle. So most of the carbon is lost as carbon dioxide, okay? So this is not very efficient in terms of carbon. So in comparison, E. coli would have a 30% carbon loss. So this is a very high loss. So we should think about engineering strategies where this vanillin can be diverted into valuable chemicals, but instead grow this organism through other valuable or through other cheaper carbon substrates, right? So to confirm our findings from flux analysis, we did some transcriptomics studies. So the bluish in color indicates that the genes for that particular pathway have been overexpressed more. And as you can see here, in the presence of vanillin, some of the genes that are in the vanillin catabolic pathway genes are overexpressed, confirming our findings that the vanillin catabolic pathway has the highest flux, followed by the TCA cycle, and plus the trans-hydrogenase genes, which convert NADPH to NADH. As the vanillin catabolic pathway produces more of the cofactors, NADH, and this pathway is important for the production of NADPH as well in the organism. And especially in the context that we also understood that this organism does not have a functioning PP pathway, right? So we went ahead and did some pH studies with this organism. So we saw that this organism prefers alkaline pH. So this y-axis shows increase in growth or cell biomass. So as we increase the pH, we can see that more cell biomass is being accumulated, confirming that this organism prefers alkaline condition. Therefore, we can utilize alkaline pretreated hydrolysate for valorizing aromatics that are present in there, right? So far we have shown studies to understand the systems biology or fluxomics in the organism of interest. And now we want to show a case study where lignin can be depolymerized and valorized directly. Okay, so we wanted to develop a strain that can secrete enzymes that can break down this lignin and release some of the valuable chemicals such as benzaldehyde, vanillin, and so on. Okay, so we engineered such a strain and shown over here results of gel permeation analysis. And over time, okay, so in brief, what happens is as I analyze my sample in this instrument, so at initial times, high molecular weight compounds are released. As I analyze it at later times, low molecular weight compounds will be released. And the comparison is the between the engineered strain and the wild type. The wild type here is the negative control ABNE. And as you can see here, compared to the wild type, the engineered strain accumulates more of the low molecular weight compound. Okay, and so this confirms that the engineered strain is able to depolymerize lignin directly. Okay. So we analyzed the supernatant that is released out of the engineered strain via GCMS, and we can see the presence of different important aromatics that are present. Okay. So it brings up the point or the case that I have made before that lignin can be valorized directly to produce a replacement source for different chemicals that are found around us. And listed over here are the different uses for the chemicals we can see here. So they can be used as a flavoring or fragrance or resins or perfumes and so on. Okay, and they also have pharmaceutical uses. So with this, I have demonstrated how we could valorize or how you could use a synthetic biology to valorize the lignin component of plant biomass. And next, we will show a technology that could be used to valorize all the components that are present in plant biomass directly. Okay, so this will be the last section of the talk. Okay, so in here we utilize a cell factory called Corynebacterium glutamicum. Okay, so as we would see later, along with utilizing sugars, C5 and C6 sugars. So this organism can uptake aromatics as well. Okay, so we could engineer this organism to convert all of the monomers derived from plant biomass into a valuable chemical. And in this case, we still focus on D-lactate, which can be used for biodegradable plastics. In short, a mechanical and a chemical process is utilized to convert corn stover into the monomeric units, glucose, xylose, arabinose, and aromatics. And this hydrolysate is fed into the engineered organism, and we want D-lactate to be synthesized. Okay? So this engineering was done by my previous postdoc, Amit Jha. So he engineered this Corynebacterium glutamicum to produce D-lactate, and also overexpressed the genes that code for the assimilation of arabinose and xylose as well. Okay? And along with that, we know that this organism has a capability to uptake aromatics. And my student Apur Matre, he went ahead and did some 13C fingerprinting to understand the pathways by which the aromatics can be assimilated in this organism, or to confirm the different rate at which different aromatics are assimilated in the organism. Okay? So he fed the organism with labeled glucose and unlabeled aromatic carbon substrates. Okay? So this y-axis shows here the labeled fraction. So if the biomass is synthesized from the labeled substrate, then this labeled fraction would be high. So this is a control. So the organism that was grown only in glucose, so it has a very high bar. But in the presence of unlabeled aromatics, this bar, the height of the bar starts going down. Okay, and especially in the presence of fumaric acid, so this building gets a little shorter. Okay, so indicating that this organism prefers, out of all the aromatics that are being fed, this organism has a preference towards fumaric acid. Okay, so these knowledge would be valuable in engineering this organism for different pathways. Okay. And plus, this confirms that along with taking sugar molecules, this organism indeed has aromatic assimilation capabilities. All right. So we engineered, or we constructed this engineered strain. We conducted initial studies. Later on, we collaborated with a collaborator at Sandia National Labs, Dr. Davis. So he works on making those hydrolysate, or he works with this collaborator to making this hydrolysate, and we conducted the bioreactor studies by utilizing the strains that was converted. And we know that this organism can decrease glucose and convert into lactate. We saw about 12 grams per liter of lactate production. And importantly, there are several aromatics that are present, and we saw their levels going down as well. Okay, so with this, we show that we can use this organism and convert all the organic carbon substrates that are present in plant biomass into an useful chemical. Okay, so with this, I end the research part of my discussion. So I would want to thank my PhD and postdoctoral advisors. So a couple of talks that was presented here came from there. And along with that, I would also like to thank my research team. So apart from different students and postdocs I mentioned about, so we have a large pool of undergrads and a large pool of hardworking undergrads. Okay, so they also form a foundation of our lab. Okay, so I would also want to thank all of them, and importantly, all the funding agencies without which we would not be able to do all these, highly expensive research work. Thank you all. And thank you all for listening. I think must have been like 8 or 9:30 there, right? Okay, so thank you for the ones who are staying late to listen to my talk. Questions, if any.
Thank you, Professor Arul. Varman, such an interesting talk. So many things. Really, I enjoyed the talk. It has so many content. So now the talk is open to question. Like the most important thing, you have used the 13C fingerprinting for all of your strain study. So have you estimated the fluxes or it is impossible to measure, calculate the fluxes? Like, have you fitted the metabolite labeling with experimental theoretical labeling to get the fluxes?
Okay, good question. Okay, so fingerprinting is a bit more easier to do, right? When you
Yes.
compare to flux analysis. So as a preliminary study, we did fingerprinting for most of it to understand the pathways. But as you would understand, the funding is the constraint, right? So we get some initial data, and of course, if more funding is available, then we would be interested to do flux works and compare the systems level changes between the organisms. Yes, we definitely have to flux studies for the different organism, but it's the funding that is the limitation. Okay, so there is only certain works we could do, and beyond that, to invest, we have to think about funding. Yeah, funding is a limitation.
And during your initial talk, you spoke about the strain. You have a consistency of engineer strain, and then you are selecting the best strain out of it, which is consuming, which is breaking xylanase into the enzyme which is secreted by this microbe, and then you are selecting the best organism, which is actually converting xylanase to monomer sugar like xylose and other things. So is this engineering? How you are doing the engineering? Like, you have not told about how you are engineering this.
Okay. So the reason, yes, the student did catch up with it, caught up with it. So so the reason I haven't gone into details is I didn't want to put oppose a paper that is still under preparation. So I can tell it in brief. Okay, so along with overexpressing genes of interest, what we have to do is with that gene, we have to attach what is a DNA sequence called as signal peptide. Okay, so this sequence will say this or this protein needs to be secreted out of the organism. Okay, so that's a synthetic biology technique we have to we take advantage of in our laboratory to secrete different enzymes. So we engineer a bacterium again. So the host selection is also important in metabolic engineering and synthetic biology. So people were asking about different things you have to focus upon to do effective metabolic engineering. Host selection is important as well. So since this work is unpublished, so I haven't described the word host that was utilized here. So the particular host was selected for its very high enzyme secretion capabilities, and it was engineered to with this signal peptide along with the gene of our interest, and it secretes the enzyme, right? So you can look at the literature for what is called as secretion pathways. Okay, so you can look at the secretion pathways that will describe in detail how the proteins that are synthesized. I'm telling to the student who is asking that question. Okay, search for secretion pathways, and you can see you can look at the machinery that is involved in the secretion of these proteins in detail. Okay, so in essence, we will you have to attach what is called as a signal sequence that will signal that this amino acid, or sorry, this protein needs to be secreted out. So I can secrete cellulase or xylanase in that particular work, and ligninase in the other work, and we could talk about several different polymers as well.
So in the cyanobacteria, you have synthesized the product where you have used the leveling of glucose, and as well as have used carbon dioxide without labeling. So you just use one carbon level, not fully labeled glucose you have used. So if you are using less labeling of carbon, will it affect the carbon labeling, or when you, generally we do 80% labeling, like 20% one carbon label and 80% fully label for our yeast experiment. But this time I saw that you have used only one carbon level only, and then what is the ratio? Is there any fully carbon level in your mixture or not?
The substrate was not, fully carbon labeled. Okay, so in this case, this experiment, this strategy by itself was sufficient to say whether the labeled carbon from glucose went through the metabolism or not. Okay, so we just want to infer that, and for that, okay, so this is more about availability. Okay, so like you rightly said, if we used a fully labeled glucose, it would have been better. But what we had in that lab at that time. Okay, so we had this. We either had first position labeled glucose or glucose with two carbon labeled. And for our particular experiment, this was sufficient to interrogate, and we went with it. And it, of course, we can see the labeling penetration into the metabolism, right? So there was no need to say, okay, we have to have fully labeled glucose in there. This was sufficient. So we we made use of available resources. That was the reason. Otherwise, the perfect experiment, like you said, would have been, okay, go with fully labeled glucose. So there wasn't any technical reason. Okay, so it just it was resource availability.
Yeah. Yeah, there's a lot of things you have explained. Really nice talk. I really enjoyed. I have to explore more. I have to understand. Many new things came up in your talk, which I have to learn. I really enjoyed the talk, and I think we have no question from the student, right? So we may conclude now of this one-day workshop. We have been watching lectures for more than 12 hours now. Everybody is exhausted. Like a lot of people attended in the morning session, which went for 6 hours, and then there was a break for lunch, and then we came back again and keep on listening interesting talks. So it was a very nice day. We get to know so many things about metabolic engineering, and and a lot of things we came to know today. So if we come in person or we have a offline workshop, then we would learn more. We actually planned a workshop in May, but it did not happen because of the current situation. And it would have been better, like if we meet and arrange a workshop, then many things we can understand more clearly. Because of shortage of time, we have to suppress everything together in one day. So many students could not absorb because this is so many things in one day. So more things will come up once we have a offline workshop, which will happen next year, and hopefully we'll meet Professor Arul, Varman, and then other faculties like Professor Tang also interested to join next summer. So to make this thing happen successful. So Arul, you want to add anything and then conclude this workshop?
Yeah, thank you, Amit, for conducting this excellent workshop. So, like Dr. Ghosh mentioned, so I'm highly interested to visit IIT Kharagpur, and looking forward to the opportunity to be there and meet all the students and faculty at IIT. Thank you, everybody, for listening.
Okay. Thank you, Arul. Thank you, everybody who has attended this workshop. We had a wonderful time today. Even though you are not meeting personally, but we really liked the interaction we had. A lot of questions came during the lectures. It was a really motivating lecture. Everyone gave very good lecture, and several speakers spoke about different topics, and it was really a motivating workshop. So hope we will meet very soon in person and continue our understanding about metabolic engineering. Recent advancement, what is happening in metabolic engineering in terms of different microorganisms, which is very difficult to actually grow and then do engineering. It's not easy actually to engineer this cell. First of all, you have to grow that cell in the lab. And when you understand the physiology of the cell, then you understand the molecular biology of it, and then you establish the metabolic pathway and then characterize it. There are so many steps. It is very time consuming when you work on a new organism. especially Professor Arul has worked on many new organisms, and then they characterize, they engineered that, and they also measure the metabolite labeling. Is a lot of work need to be done. Professor Arul's group has done, where they understood the biology of the new microorganism. So this is really an interesting talk, and we will continue listening to him in future as well. Hopefully, we'll meet in person as well. So thank you all for all the members who helped in organizing this event, and we will have another workshop next year. Thank you all.
Okay, thank you. Bye.
Thank you. Bye.